Passing 60 meters altitude.
Looks like Sirius now is good altitude lock, 80 meters.
Understood. That agrees.
Yeah, sounds right.
Okay, yeah, that's definitely a hard target. 60 meters out. We're at 52 altitude.
Nice.
Coming back to center.
How do we feel about a bailout of 020?
We probably maybe slow down here. I think with the heaves.
Yeah,
I think it's
slowing down.
tugging Sirius a little bit. Nav, you said 030?
020. We could do 30.
I think that general direction sounds good to me.
Yeah, I think just higher up 30 is kind of 20 cuts off that slope a little more.
Sounds good.
All right.
Yeah, would you be able to range our blue view into 60 meters now that we're within that threshold?
Yeah.
Thank you. Coming back to center, pilot.
Copy that.
Altitude of 38.
Good.
Down between my toes with the main HD. Blue view's looking down. I'm going to speed up a little bit.
Understood.
Came down quite a bit. Delta of around 14. 12. Back to center. Thank you, Nav. We have blue view readings under 40 meters now. Altitude 31 meters. Those numbers would agree with the 15 to 20 degree slope.
Sirius altitude 45 to 25.
Copy that. And up to 20 meters altitude, should start being able to see visually, depending on the turbidity of the water. $15. Slow down a little.
Copy that.
Okay, I have visual at the bottom. Coming to 10 meters altitude. I'm slowing down.
Understood.
Good morning, Deep Sea Explorers. After 2 days away for weather, we are back in the water and exploring Gosnold Seamount this morning. I'm just going to let the pilot set up here. We've just had the bottom in sight, and then we'll begin our dive.
Our Z-bias is pretty high. Continuing down. I'm at a 175 right now. What did we say, 190? Is to waypoint 2, but what is
I think whatever you think.
slope?
Yeah,
Slope
whatever
is,
looks
yeah.
good for local slope.
I think 175 looks pretty spot on. Okay, gonna start lifting up. Come down a little bit more.
Alright, I might pause around there.
Copy that. Continuing down.
I can start in your swing arms if that's alright.
Yeah, if you don't mind. Thank you.
Do that. Hydraulics are on.
is an interesting bottom.
It's nice, flat, and hard.
Perfect landing site.
And pipe, be advised, as if layback adjusts, I'll be moving laterally relative to you.
Understood.
Great. Bridge, RV nav. I wanted to let you know that vehicles are on bottom, and I wanted to verify center of rotation is set to A-frame.
That is a
Great,
flat bottom.
If now would be a great time to talk about a bailout plan. Okay, perfect. We're looking at a bailout northeast bearing 020. Copy that. Thank you, Bridge. Go ahead, Bridge. Good copy. Thank you, Bridge.
And pipe, be advised, you're a little bit out there.
Yep. Good for that.
Not a danger, but you're not going to be able to make much forward way.
Yeah. I'll come back a little bit to center. Just trying to get in Z-bias.
Yep. Test out auto XY too. Nice flat bottom. It should hold good here.
Yeah.
All right, lower swing arms. Going outboard and deploying. All right, pilot lights are out. HP is
Understood,
off.
thank you. Jeez.
I guess, let's see, I'm looking, I mean, 175 looks like the right way. Do you want to, jeez. Do you want to start with 30 meters and see how the vehicles react?
Yeah, we
Go
can
from there.
We can do that. 175,
see.
30 meters.
Yeah. Oh, and you can talk with Washley and see if they want anything before we start moving.
Yeah, just trying to get.
Yeah, when you're ready.
in.
Yep.
He's pushing down like negative 31. It's still not balancing it out.
We added
We added
10
weight, right?
pounds, yeah.
I wonder why there's so much up force.
Doesn't look like a lot
I
of.
mean, unless the current's coming directly at me and kind of getting stuck in our brow and pushing up.
There could be some hydrodynamics like that happening. What is the current doing?
Geez, this is really flat and barren. This is wild. Alright. I do not have. Watchley, this is Pilot.
This is Watchley.
I think we're ready to go ahead and start the dive. Is
Oh.
there anything in this immediate vicinity that you see that you want to take a look at, or should we just start heading towards the slope?
Yeah, so we have a bit of interest in sitting down and kind of poking this to see whether this is solid pavement or whether this is looser. So I don't know if it's possible to set down. I saw kind of two larger boulders too, kind of maybe around those, just to kind of have a poke around
These
before
right
we
here?
get on.
Sure. Is this what you're talking about? These two in front of me now?
Yeah, yep, right in front there. Either one of those would be great to. We suspect this is completely solid pavement, but it would be interesting to see if that's true.
Looks like we might have a nice
Yeah, I
cobblestone
don't know
road
if D2 has a
brings us-
jackhammer of some type, but
I'm not sure they put on the jackhammer attachment this morning, Jason.
yes, this looks like a nice cobbly road here.
Let's see.
Yeah, I think this is
Dogs
solid.
up.
Good for that.
Botryoidal ferromanganese pavement,
It's,
which we have not seen anything like this before.
it is really extensive too. I mean, this just goes on and on. It's really quite fascinating.
There is a target over here to our port sticking up. I don't know if that's of interest. Alright, arms coming live. If you feel comfortable, co-pilot, if you could
Yes,
bring up Mini.
of course. Monitor right, going to HD2. I'll bring it down.
Alright. Arms coming live, arms on, arms coming out. And you were just looking for a kind of a poke test, Watchley?
Yeah, kind of a poke test to see whether anything is actually loose around here, whether this is all completely paved over.
The focus being this very spherical rock.
That one's pretty solid.
I did see it rock.
Actually, yeah, I might be able to grab that watch lead.
Oh, really?
That's
Yeah,
of interest.
yeah, that would be great.
Did it move?
I think so.
It definitely shifted a little bit.
I'll center up
Copy
camera
that.
here.
Alright.
Do you want
If you
the,
could zoom in on the main HD.
Do you want the lid open first? Or do you
Yeah,
wanna
we could
go
go
other
ahead
side?
and, I was thinking the starboard, but we could put
Oh,
it
you
in
can go
the...
start, whatever you, yeah. Starboard's totally fine.
I did get a scratch on it. Let's go and pick it up. Alright. Come out partial zoom video, please.
That is definitely a ferromanganese coating on there.
Hanging out on it.
Did you put lasers on it also?
Lasers are on now. If you want to come out so we get lasers, actually, they're already there. So, about 15. Alright. Let's grab this guy.
Oh, it is loose. Look at that. Twisted.
Stand by
Oh,
for
that did move.
And look at that. The nodules around it are moving as well, Jason.
These are ferromanganese nodules.
Ooh, that's precarious.
Whoa, look at that.
Alright.
Ready to wing inboard.
That is a
when you are.
bowling ball sized ferromanganese nodule, or at least a nodular shaped. Obviously, we want to be sure. Oh man, that is wild.
Yeah, we can collect that one, pilot.
Okay.
Thank you.
Yes, please.
Ready
Alright,
for
let's
wing?
go for, starboard.
Alright, that's it.
We were certainly not
Want some light
expecting
where
that
you
to be
Pressure pretty good there.
loose bottom.
Okay, coming up over the jaws. Yeah, that is interesting.
That was a bowling ball.
Do you want me to kind of play around in the hole I just
Of
made?
course.
Or?
That would be,
Certainly a bowling ball.
yeah, that would be great. Thank you, pilot. We just want to see, is this all loose? And
Yeah.
it certainly looks like it from here.
Zoom first, probably before it gets more dusty.
Sorry, say that again.
I think we
Yeah,
might
it'd be
wanna-
great to kind of poke around
certainly.
and see how loose this is.
Okay. Do you
Thank you,
want
pilot.
to come in on the hole I
Yeah,
made?
some of these other ones are
might wanna
loose.
look at it first before
We might
get
want
more
to just grab a
smoke.
couple of little ones too.
Did you catch that,
On the
pilot?
same ball.
We may want some little ones as well.
Yep, if it's possible.
I think suction sample, co-pilot.
Oh,
Depends on the density, but yeah, they're too small for the.
Yeah, for the draw.
So I would say suction sample. Worst case scenario, you might be able to scoop some in.
Yeah, that looks pretty soft.
yeah, look at that, rolling away. My goodness.
Yep.
Yeah, you might
This
wanna
one
do
here
it.
might come. It might fall down. Sorry, say that again.
Now you
Video.
might want to stir it up with your claw or something now.
Alright. We'll come in, stir it up a little. Alright, so what I'm going to do is reach in and then I'm going to pull back. Oop, that one did go. Just kind of drag.
Yeah.
Yeah, it's all
And look at
very
that.
soft and loose.
Very, very loose. Is it possible to grab a handful of those?
I'm not sure. I think we're going to have to suction sample it. We can grab some, but we'll have to bring out the suction sampler
Okay.
and pick it up.
No, I think that would be fine. Thank you.
Understood. We
Alright.
also have a fish down here as well.
Let me know when you're ready for
Yeah,
drawer.
if you want to bring the door out.
Extending drawer. That's good there.
Thank you.
Then let's see, you're on jar 6, so I'll put you
Probably go for
just one.
one.
right?
Is yeah, one's the one that doesn't have a filter.
Was that container one?
Yes.
Alright. If you want to bring
Ready
the
for
drawer in,
retract.
Yep.
Retracting.
hold there.
And Cirrus is moving a little bit. I think you're still okay.
Copy
It's
that.
nice and flat. I'll keep an eye on it.
Thank you.
Can we get the jar light on?
Yes. Let me know when you're ready.
Yeah, we're gonna try and scrape this edge here. Okay, if you want to bring on some suction,
Want to zoom?
if you could zoom in on Mini, that would be good. If we could focus on the left-hand side of the arm, I'm gonna try and bring it along the outer cusp of this hole I made.
Just 1 second, I gotta trigger the iris to get more iris.
Copy. The hole, we have good visual of jar number 1.
Looks like I'm maxed out.
I mean, some of these are. Is there? So watch lead. I understand we want to grab some small ones. Are we looking for as small as possible? Should we change to a jar that does have a small filter?
No, I think it should be fine without the
Okay.
filter. Yeah,
Alright,
I think
understood.
they're bigger,
Alright, ready?
bigger than that.
Yep.
Thank
Arm
you.
is live. Ready when you are, pilot.
Enabling suction now. It's 1000 RPM.
Copy that. I'm
Might
just
have to
gonna
give
do a
more.
little ice cream scoop.
They're going, yeah.
There we go. How many of these are we looking for?
3 or 4 will work.
Well, we have.
Yeah, 1, 2,
I think that's
Three.
We have a lot of more.
4, 5, 6, 7, 8. Yep.
All right.
Let's call that good.
You can come up with the suction.
Thank you so much.
No problem.
off.
There's a few in there for you.
That's perfect. Thank you.
All right. If you could
Those
come
will
out full
be
wide on me,
used
please.
extensively.
And if you want to come out with the drawer, co-pilot.
Extending
That was
drawer
jar
now.
1, correct?
Yep.
Yes.
I'll center up this camera.
Copy that.
Stop there.
Well, that was quite the find on the landing here on the Seamount today. So good morning, deep sea explorers. Today we're exploring Gosnold Seamount, having had 2 days out of the water because of weather. We're on dive 16 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. And today we've landed in about 3,200 meters of water, and we've landed in a nodule field, which is very, very unexpected for this area.
There we go. I was getting a little confused with that. Okay. Arm is clear.
Retracting.
We'll take another tight look at the sediment while we're here.
Yeah, let me just stow the arm.
I rotated 10 degrees with Cirrus. I still think
Understood,
you're
thank
good,
you.
but just be advised.
Okay. I'm secured. I'll let the, you feel safe, co-pilot?
Yeah, I think I'm still good. Tether looks nice and loose. I'm just laterally moving here.
Copy that.
Still good view and aft camera. Sonar looks clean.
We'll let this dust settle for a second, and then we can come back in and look at the ground if that's of interest. Watch lead.
Yeah, that would be great. Thank you.
Do you want blue
Can I
view
have a
back
little
up?
scan around?
Yeah.
It's not super helpful with all the dust.
Thank you.
Do you want me to zoom in to see what I can make of it?
Oh, yeah, you can try and start coming in. May not be ideal, but did seem quite fine underneath this top pavement layer.
It'll tilt down.
Coming down a little. Holding there.
Hydraulics coming off.
Copy that. Thank you.
HPU off.
Yeah, we have this thing over here. Not quite sure what that is. Off to the port.
It's kind of, yeah.
30 meters out, though. It's in the wrong direction, right?
Yeah, and you have to come this way to center with me too.
Yep, understood.
Once you're done,
Video's
you're fine
clear.
now.
Alright. Watch lead, we'll continue on.
Yeah, that would be great. Thank you.
Look at that. Coming off.
So yeah, I say we're gonna move ahead, and you can just center up as we move if that works.
Pilots, I need to do another black balance on the camera.
Okay, I'm in a safe spot if you want to go ahead and do that.
Capping the camera.
Understood. Watch lead, we're just doing another black balance check just
Okay.
before we continue.
Perfect. Thank you.
Pilot, I'm on George 6 now.
To everyone at home, they're just testing out the cameras before we
Copy that.
proceed on our dive here. Just getting them balanced and ready to go.
Go ahead, watch lead. Sure. Yeah, go ahead and do that. I'm gonna come center myself up, pilot, co-pilot.
Understood. Suction's disabled.
It's much better.
Excellent.
Oh.
Speaking of that, there's a fish.
And do you want to get that moving, pilot?
Yes, hooray for the
Video's
move.
clear.
Okay.
If you want to come in on the fish,
Yeah, 175. Do we have a distance we want to go?
30 sounds good to me. I think.
30. All right.
Does that work, pilot? 30
Yes.
Looks a bit like
30
a
meters.
Snaffabranchid eel. We've seen a couple of these on previous dives here.
lasers
Bridge
are on.
RV nav.
Typically have all these scratches and
Yeah,
scrapes
it's not a
along
good
their
time to
body,
put in a
both
ship
from
move.
predator-prey interactions,
Great,
but
we're
also
looking
from
at a range of
trying to find
30
prey in amongst
meters,
larger rocks.
bearing 175 degrees at a speed of 0.2 knots.
Pretty hard to find a much larger rock here
That's
in
it,
this
thank you,
extensive nodule field. Barely even room for the sediment to poke above. The nodules are essentially blanketing the entire seafloor here.
Nav, what's the layback right now?
And sitting in, the nodules appear to be sitting in the sediment, at least
193
partway, and then the tops are
That's layback
poking out.
for first move. So we call that
We have
starting point to reference for this
several ferromanganese crusted nodule experts, and Kira Mizell, who was with us just a few days ago, is one of those. And
Squad logs on the left, crinoid. in the
we're
south.
sending them information as quickly as we can to find out more. I was not expecting, and this is Jason Chater. I'm the research geologist at the USGS and the geology science lead for this dive and those to come. This was definitely not on our expected list for this dive.
Sorry, say that again,
Can we
video.
get up real close to him?
Yep.
Yeah, this was definitely a bit of a surprise this morning. landing in a nodule
ton of
field.
tether,
We're
but-
diving today kind of in a saddle between two peaks of Gosnold Seamount. And honestly,
about
talking
it.
this morning,
That's
we were
about what we
expecting
got right
maybe
now.
some sediment or some swept rock.
I gotta
And
move
here we
this
are
way,
in a ferromanganese nodule field.
If you rotate
And
this
here's a-
way,
Rian,
you might get
sorry, if I
Yep.
can interrupt for just a second. There were at least 2 or 3 bamboo corals around that
There-
rock that we've
Yeah, it's
been
about
on.
what we had.
Yep, there are quite a few in front of us too here. So hopefully we're going to take a closer look at those in just a moment, Scott.
Sorry, what are you looking
Thanks. You're still
for?
getting stable. Sorry, thank you.
Sure.
Hey, bot, you're gonna have to back up to me. I think I'm still moving,
All right, stand by one.
and I'm just getting pulled with tether right now.
Okay.
Kind of see where centered, and I think Sirius is just keep moving that way.
Yeah.
Yeah, it's better.
Right, video if you want to come in on the squat lobster.
And here amongst these ferromanganese nodules, we have a squat lobster. Very often we see
Copy.
these squat lobsters hanging out on rocks or even on corals and sponges. And they tend to pick out particles, larger particles, either out of the water column or between rocks for food. Scott France is just noting that this one actually has potentially embryos underneath as well. Beautiful orange eyes on this particular squat lobster.
Anything in particular you want me to do, video?
No, that's the best we can do.
See the antennae
Nice to be
up
closer
in the
than
water
we're
column.
on
Those are
tether.
kind of tasting the water, looking for food.
We're just waiting for tether. And then you wanted to look at this coral down here?
Yes, that would be great. Any of those, yeah. One dead center would be good.
Don't
In
know if
amongst
we'll
all
be
these
close
nodules, there
enough
are
to
these
it.
thin corals sticking up. And they're very, very small, so it's going to be quite tricky to get a good look, but looks like all the polyps are actually in as well. I'm not sure if Scott France is on the line.
We're gonna have to get a lot closer to see those.
Yeah, I'm here, Rian. These are
Sure.
Primnoidae
Yeah.
corals. I can see
There's
the
a lot of
more
current
tether.
right there.
scaliness
All right, I
of the
can stay
polyps.
closer
And
if
the
you want
polyps
to
are
come out, video.
rising in
Away.
small whorls. I'm not sure what genus. I know that there is a genus Candidella that forms these little primnoid whips,
I'd hold that.
but
Okay.
I'm not certain. They're very small. There might be a bryozoan or a sponge also in the foreground, but there's
video
plenty
if you
of
wanna
little
come in.
stuff growing on these nodules. Very interesting.
Indeed there is. Yeah, indeed there is. I think, we've seen some forams and then potentially some bryozoans as well. In the background there, I think there's some small sponges. There's definitely a lot of small fauna here. And a lot of these smaller little potentially primnoid corals as well. As we're kind of zooming in and backing out here, you could see a lot of these little coral sticks sticking up from the nodules. And this one's quite firmly attached to one on the top of these nodules. I can see the base quite clearly here. Beautiful.
And tilt down to the left.
Yeah, I'm trying to continue to convince myself that I'm really looking at a primnoid and not a little sponge.
You see
Actually.
something else
Oh,
right here?
interesting. No, I think it's a primnoid. I can see
Saw a
polyps
soft
quite
coral
clearly on
left,
the
upper left edge.
high definition screens that we have here. Kind of looking all around here as well. Oh, what's this one? Oh, this looks like a soft coral. Yep, you can see the pinnules on the tentacles there.
Go ahead, Bridge. Copy, thank you.
Very well disguised
Shipment is
in
complete.
amongst this
Cover that now, thank you.
nodule field here.
Yeah, for sure. I agree with you there. No axial skeleton to this octocoral, just basically fills the colony with water, can channel water in and out to change the size somewhat, make it compress down to the rock, or to get a little bit taller for feeding.
Yeah, and then Megan Putz is, Megan McCullough is saying those branches are little branching white bryozoans as well.
Actually, I think they said the squat lobster had potential embryo underneath it. Maybe we can get a look from the front.
Yeah, we can see if that orange is embryos underneath there.
We are pretty close. It's going to be right between the toes. Come in.
Good morning, everyone. This is Kira Mizell tuning in. I was woken from a slumber to check out these nodules. Wow.
Good morning, Kira. It's great that you can join us this morning, even though you are officially off duty today. We've managed to drag you back in.
I hope this is a good thing to wake up to.
It's interesting in the back
Absolutely.
as well, like the sea floor. Everything's covered in this.
That's great. Thank you so much, Pilot, for that beautiful imagery of that squat lobster there.
Right. Let's continue.
Yeah, so it's really, regarding the nodules, if I can just chat about them a little bit as I've gotten oriented checking out the map where we are. It's incredible on that zoom, too, seeing we've got all these nodules as well as ferromanganese-coated debris of many other kinds. Seems like what's happening in the saddle between these two seamounts, this is just an amazing coverage. So nodules, they require a nucleus to precipitate around. And so this is showing us that there's an abundance of material here that can serve as a nucleus to form these manganese nodules. Unlike this isn't quite an abyssal plain region where you could, have a deep seafloor that would have nodules sitting on sediments and might be able to get metals as they grow from both the sediments and the seawater. It appears here in this saddle that they likely would have a similar composition to crust on any big outcrop, but really just highlighting the vast amount of material that's available for these nodules to form. This is just a spectacular site. I'm glad you guys picked up a few so we can check out the chemistry and see if these are forming mostly from seawater and are an interesting analog to crusts in this region. It also, ferromanganese nodules need a really low sedimentation rate in order to form, which is why they tend to form in parts of the abyssal ocean where there's not a lot of surface productivity to have that marine snow falling on them. So it's also a good ode to the current strength here, that they've been able to be swept free of sediment long enough to form. They're also quite rounded, which the reason that manganese nodules get rounded is a bit of a mystery, and how they stay at the sediment surface as well. Is it fauna pushing them around? Is it they're light enough to be pushed and rolled and they stay rounded? So this is just incredible to see this density and this rounded atmosphere for these nodules. It's just incredible. So that's my long spiel. Happy to stick on the chat and keep looking as we continue forward. But thanks so much for calling me in. This is wonderful.
Thank you so much, Kira. Really appreciate that download on what nodules are. It's really interesting to see all these organisms as well that are living in amongst these nodules. At the moment, we're looking here at a stalk crinoid that has a bit of sargassum that's been wrapped around it. So the sargassum grows up in the upper water column and eventually becomes encrusted with so many organisms it sinks to the seafloor. And here one has got caught up on this crinoid. I was wondering what the orange was there. Thank you so much, Pilot. If we could zoom in on the sea star that was up in the foreground, that would be great.
Yeah, video. All lights are as dispersed as I can get them right now.
Okay, coming in on the sea star. Go ahead. Six-legged, huh?
Yeah, so this is a sea star. I'm hoping Chris Ma is on the chat or on the line here. This is not one that I recognize.
If you want to, I'll pan down. We can take a look at one of these.
You can see the long tube feet of the sea star. These sea stars are echinoderms, so then the same phyla as sea urchins and brittle stars, and that crinoid that we were just looking at as well. And these sea stars have this water vascular system that controls the arms and the tube feet. You can just see those tube feet sticking out there. Christopher Ma says this is an amphoraster.
I just want to point out to you, just to its right, I'm not sure if that's a cup coral or an anemone.
Yeah, and on the left as well, there's another one of those soft corals as well. There's actually, you know, every time we zoom in here, there's quite a bit of life on these nodules. Yeah, so Chris Moss says amphoraster for this particular sea star. If we could zoom in, pilot, on the kind of the top right of the sea star, there's tentacles sticking out, kind of a reddy color.
Go ahead and come in. There's two of them. You want the upper or lower?
Oh, yeah. Actually, the lower. So the upper is another one of those soft corals we were just kind of looking at. And this lower one.
That's full zoom video.
Yeah. Really tucked in between all these little nodules. It's hard. Yeah. Unknown if that's a little anemone, a cup coral, or even a cereanthid that one could be, and we're just not seeing those inner tentacles.
You can get a little bit closer if you'd like. We do have some more tether, if that's of interest.
That'd be great. Thank you.
All right, full wide video. I'll scoot in a little bit more. Looks like we have another one of those same sea stars up upper right.
So the organisms that live on these...
Hey, Rhiannon.
Oh, hey, Chris. How are you?
Good morning. This is an X-rayed. So this is a pedicelasterid. This is actually kind of interesting because these are distant relatives of the inshore, like the really common Asterias, except that for that whole group of sea stars, these occur down at the bottom of the tree. These are sort of very early forms relative to the things that everyone sort of looks at, you know, when you're looking at mussels and oysters. And some of the fossils for the animals that are like this occur from the Jurassic. So this is we actually collected one or two of these before, and they're extremely variable. It's really interesting how different they look relative to the preserved ones. And so I'm really happy to sort of just see more of them. The weird thing is that this also could be a bizarre related animal called Polystereus, which is something that we saw both in the northern Pacific on sort of vent habitats and in Antarctica. So I mean, it's unusual, and we don't know much about them, and so I just feel the need to call in and register my appreciation for seeing one. So I'm grateful for you guys to stop and look at it.
Yeah, I think there's actually another one coming up here, Chris, as well. I can see very distinct pink arms. I don't know.
Yeah, at first, was it a squat lobster?
No, I think this is a sea star in a feeding position, so it's got the big bulge in the middle. It's kind of hunched over something.
Your move is complete.
That would be nice.
No, I don't think I am.
Okay.
I think I'm still moving a little bit.
Understood.
It's being a little funny though with the layback. But I think we could get another move in.
Copy that.
I think it's about.
If you want to start coming in video, this looks a little odd. Different coloration. All right, sorry, there's something in the way.
Oh, it's got some more of that sargassum wrapped around it as well. But yeah, it's in that feeding position, Chris. It's all kind of hunched over something under there.
Well, let's see. Maybe it's just feeding on the sargassum. Oh, neat. Okay. So they're feeding on something. Yeah, so this demonstrates one of the things about Okeanos Explorer that I love about the program is that we can actually make observations of these species doing what they do in the wild. You know, these in situ observations of feeding and other biology, because frankly, a lot of the animals that we see, that the reason we know about the animals at all is because we see dead examples of them from trawls, and so there are fundamental gaps in our knowledge. Everything from the color to how do they feed, what do they feed on? I mean, even Brisinghids, which everybody now recognizes as holding their arms up into the water, the early hypotheses regarding when they first collected those, people thought that they just dragged their arms along the bottom and caught prey with their spines. And that's very different from what we know today. They only started seeing Brisinghids with their arms, held up into the water current, I think in the 70s, when Alvin started going down. So even something relatively simple like feeding like this is food for the gods for me, because I can, and who knows what we'll see that, it might be feeding on. It might be the sargassum, or there might be something else here that might turn out to be a potential prey item, such as a small, it could be a mollusk, or it could be one of those bryozoans, or any number of other things.
Yeah, Scott.
So, hopefully, we'll get to see.
Yeah, Scott France in the chat just mentioned, of course, every time we've zoomed in, we've seen some of these small little soft corals, and we know that some of these sea stars can be predators of these soft corals, and so we really wonder if it's that. Beautiful.
That would be interesting. Well, let's see. Anyway, I'll be watching. Thanks very much.
All right, thanks so much. So this is really interesting. So this is a Pagarus, Parapagarus, I believe. This little crab with a shell, and on the back of its shell is actually an anemone, a clonal anemone. I don't see the anemone out right now. All the polyps are kind of closed up. But again, in the zoom in, we can see lots of these soft corals as well. Beautiful.
Pilot's clear.
More of these primnoid little whip corals, mini whip corals. Beautiful.
And that move is underway.
Yes.
Understood. Thank you. All right.
So if you're just joining us today, we've just landed on the bottom a short while ago. We're on Dive 16 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. And today we're on the New England Seamounts. We've actually moved further west. We're kind of on the final stretches of this expedition. And we're diving on Gosnold Seamount. We're diving in kind of a saddle in between two peaks where we expect it to be relatively high current. And our depth today right now is 3,236 meters. And when we landed today, we've actually landed in a ferromanganese nodule field, which is very, very unexpected for this area.
Take it.
So Jason, do you want to give us a little rundown of how these nodules are formed and why it's pretty unexpected that we found this today?
Hopefully I'm not too drowned out by noise. It was extremely unexpected, and unfortunately the biology is doing its best to upstage this really awesome, even wicked awesome start to the dive for the geology. But for those who've been following along with the dives so far, we have talked a lot about these ferromanganese crusts and pavements, and we haven't talked much about nodules. And is that an enemy? I'm just going to ignore it.
It's an enemy. Don't you worry. I'm writing these down. Keep on going, Jason.
So, we haven't talked much about nodules, and that's because, nodules require, a specific set of conditions for formation. And Kira Mizell from the USGS Santa Cruz office and is the co-geology lead on this expedition. Talked a little bit about it. So, getting the right conditions in the right spot is important. And because we don't know very much about these seamount environments, finding nodules was not expected. And that just may be a sampling bias that we have and not fully understanding the conditions. But so if you've seen us on other labs, we sometimes try and push some of these nodules around to see if they are loose, and so far they've been primarily part of the crust, so this petroidal grape-like texture crust. They haven't been loose, and we expected that would be the case today when we poked them. But we were happy to find that they were loose. So this really tells us something about the conditions in this area. And
something up in the water.
The pilots are certainly working hard, and the ship's crew working hard because of the heavy currents in there. So they're sweeping through here, keeping sediment from accumulating. So there's water column floaty, wiggly things again.
So this is a ctenophore. Yeah, there we go. It's gone out of shot.
Talk right through it.
All right, let's take a nice good look at the bottom.
There are several ways to generate these nodules, and that's either directly from the minerals in the sediment, and that has a lot to do with the oxygen reduction environment, organic matter, and other things. The other way is through seawater, and that's we've been seeing these crusts, and those crusts are precipitated from the seawater. And then there's a hybrid way, which is kind of a mix of both. And
ship move is complete.
And so there's a lot of sort of back and forth in the chat about what is the process that formed these nodules, and certainly once we get them back, folks like Kira and others who study these ferromanganese crusts will be able to look at the chemistry and morphology and really start to figure out what the conditions are that form them, and sort of investigate them more completely. It's always exciting to find something unexpected, and certainly something that as extensive as this. So you can see these nodules are forming a almost complete blanket over the seafloor. And just from that perspective alone, it's quite amazing. We see bits and pieces of different geology down here, but to see such an extensive, deposit of these nodules and is really amazing. So that's why, from the geology side, we're pretty excited.
Yeah, very much.
I'm not. I'm not a ferromanganese nodule person per se, but I find them entirely interesting because of the processes that go into form them, and they do host, as we've seen so far. They do provide suitable substrate for a variety of benthic fauna to form habitat, and how
Bridge,
long the
RV
habitat lasts, I guess, is really dependent on
Yeah, we'd like to put in another ship move.
the processes that are had gone into the formation and continued precipitation of these paramanganese crusts. So lots of feedbacks between the geology and biology.
Yeah, absolutely. When we've been zooming in on these ferromanganese nodules, we've seen just a lot of different organisms, from these small soft coral whips that we see sticking up to some true soft corals that we've seen on these nodules. There have been isopods and forams and bryozoans, and then some of these larger sponges as well that we're seeing kind of around as well, and some of those sea stars, of course, that we zoomed in on earlier. So it's really interesting to see all of these organisms. We're not seeing many of the big forms, maybe because it's a little bit too much of a mobile bottom, where the bottom moves around a bit. It's harder for organisms to attach to, but certainly a lot of life around these areas. Hey, Pilot Watch Sleep?
Yeah, go ahead.
Can we zoom in on the crab on the very bottom right? I think this is another one of those hermit crabs, the Parapagoras, that have the large anemone on its back here.
Copy that.
It's interesting to see two of those.
Right, video if you want to start coming in.
Yes, I believe this is. And you can kind of see that rather than being able to see the shell, you can see this large anemone that's curled over. It's not open right now. This particular one tends to have several different polyps on it. And that's the second one that we've seen so far. Thank you so much, Pilot.
Sure. Right, Pilot's clear on video is. My bad. We can continue.
These larger nodules, we'll call them nodules at this point, and we did sample one earlier that was more than 10 centimeters in diameter and did look like a very impressive
you asked for a quick zoom on
bowling ball. Finding them periodically scattered around, I wonder if there's some sort of different nucleus on the inside of those, either some sort of rock debris, volcanic rock debris, as compared to what might be in the smaller one, which may, be as small as a sediment grain or, a pteropod shell or something else. So, certainly interesting to see these sort of scattered morphologies as we go along. The bigger ones aren't always as rounded. I think we got really lucky.
Video if you want to start coming on this boulder here, center.
On that first one we came across was almost perfectly spherical. I guess if we zoom in on this rock here, there's something on top of it.
So, Jason, you said that this was likely volcanic debris?
Maybe. Kind of, hypothesizing out loud on, why these bigger ones are here. And, if the nucleus is on the inside is maybe a piece of volcanic debris that's come off the seamount or something else, maybe you're able to generate a bigger nodule, so to speak. It's the kind of thing we can't see from outside. Can we zoom in on that one that's just coming into the center bottom frame? Wondering what that stuff on the top is there.
I have one on the left and one on the right.
Jason, would you like the left one or the right one?
The white one, please.
Okay, sounds good, thanks. Are you looking at the little white branch on top of it?
Yeah, there's like the white branch, and then there's these little, I don't know if it's in the sediment behind it or coming off the rock itself. This sort of brownish fuzz on the left side of it. Or I need to clean my glasses. It might be one of the...
What exactly are we looking for, Webster?
Oh, it was in the sediment, but it's good to take a closer look.
Copy.
The sediment behind it, you can see this kind of coarse, coarser than we've seen. There's actually a little, I don't know if that's a coral or something else to the bottom left there.
Yeah, there's a little octocoral there, and a little sea star as well.
Probably you have time, but I'm starting to inch closer to you. Plenty of time, though.
A little
Everything here is just so small in terms of the biology.
Put camera down to 6-0.
This is a sock coral that we've seen a couple of times now. They're actually quite abundant on these nodules. But on the front of the rock, there was something else that was really spiky. I couldn't tell if it was a sponge or a hydroid. The rock that we just left to the right. Recognizing that I'm delayed by several seconds.
Copy that.
Spiky white thing,
Ship
I believe I found
complete.
it.
Copy now.
Yep, Scott, we're going to zoom in on it in just a second.
Go ahead, video. It's a carnivorous sponge.
Scott, is that a carnivorous sponge? Looks like Lars got it.
Yeah, I'd say that is a sponge, and probably a carnivorous sponge. A very different sort of biology than most of the sponges that we're looking at that depend on a aquiferous system. That is, they pump water through their body and remove small organic particles from the water, whereas this one has its siliceous spicules shaped as a variety of different kinds of hooks.
It's full
and
wide,
it's described
right?
almost like a Velcro texture. And zooplankton can get caught up on those hooks. And what's really fascinating is that the sponge then reorganizes its body. Its cells actually move and grow over whatever it's captured and then digest it. So effectively, the sponge creates, when necessary, a small digestive space like a stomach. Sponges that don't normally have stomachs. And that's how they feed. Really fascinating. They're all related in the same family, Cladoryzidae. But there's a tremendous diversity within that group.
No.
What? I've never heard that was how they ate. That's so cool.
Yeah, really cool. They have no more remnants of the aquifer system. That is, they don't have the cells that pump the water. And presumably this is an adaptation to living in a relatively food-poor environment. Most of them that are known are from the deep sea, but there are some also in shallow water in caves, which are kind of deep sea analogs in that there's relatively food available because they're really dark.
Camera going to 8-0.
That's really cool. Thanks, Scott.
All right. Understood. Watch lead, is this a sample or are we continuing?
Let me check. Folks online, do you think that this should be a sample? I know there's not much known about carnivorous sponges in the deep sea, but are there any requests for a sample of this one?
I think it would be a welcome sample. I imagine that what we're talking about is picking up the rock, right?
We would potentially suction sampler, but let me confirm.
We could attempt the suction sampler as well.
Awfully small.
It is quite small.
I don't know what would be left after you tried to suction that.
All right, well, maybe we'll keep looking then.
Okay.
How big is that rock? The rock's definitely not going to fit in a bio box, but if you could come out until you can see the lasers video, please. Thanks. Yeah, it's pretty large. I mean, we could grab the rock and then hope that the sponge stays. But
So, folks, there is an option here to potentially collect this rock, and if we do, it would go in the rock box. We would not be able to fit it in the bio box just due to its size, because it's quite a large rock. If that is the case, though, we may risk potentially losing the sponge, but we could try. So what do folks think?
We also are going to have to move the ship back if we want to collect this.
From the geology side, given that we've already got one big rock in, I think it's in the starboard bio box, or the starboard rock box, we would potentially miss out on a bigger sample of the volcanics. So from that perspective, I think I would vote for no, but certainly if the biologists are
I can agree with you, Jason. I think you're right. I think the chances that we would lose that due to abrasion in the rock box mean that we should save the rock sample till you see something that is truly interesting to you, and we'll just leave that sponge be. But thanks for the suggestion, Casey.
Great, thank you guys. All right, looks like we're coming up on a couple more of these boulders with some sponges on them as we come into view, and more of this nodule field.
Camera 6.5 right there.
Copy that. Thank you.
Nothing on blue view, so it's pretty flat.
I'm going to do a quick snap. This might be a bottle video. I like to just see if there's a message in these.
So this looks like it might be a beverage
not.
bottle,
Right,
some
forward.
anthropogenic trash here on the seafloor. Unfortunately, we do see this sometimes, particularly as we get closer to shore and in more densely populated areas.
Another sea star for Chris Law.
Yeah, I think that might be the first piece we've seen in a lot of dives. I remember one from the first dive, but don't remember seeing anything from all the other ones.
That fits my recollection as well, Jason. As we're getting closer to a populated coastline, though, we might start to see more of this over time. So, Kira, I believe you are still on the line. You had made some observations about the nodules when we were just zooming in there. Would you mind telling us a little bit more? I'm certainly still learning about these as for me personally. I think folks would love to hear more.
Camera's 5.5 there.
Copy that.
Hi, Casey. I was just mentioning in the chat that on some of the zooms in on the sediments, which the biologists have been making some great requests to zoom in and see the smaller stuff living in and around nodules. They can create a really fun environment, being hard substrates scattered about sediments. Am I looking at a fish real quick? I'll let somebody ID that.
With parasite.
Yeah, with a couple of those parasites there on the edge of it.
Starting to get out of my view. Have to let him
I just, and so I was making the observation that on one of those zooms, checking out the infauna amongst the nodules, you can see some broken nodules that have a really finely laminated texture on them, indicating that they're likely hydrogenetic. You can still get concentric growth for all nodules around a nucleus. You can imagine it sort of growing out like a tree ring.
We can let him go.
But that broken surface sort of showed me in those fine laminations that's more a signature of hydrogenetic growth, meaning that the metals are coming from seawater and the oxides that are forming those ferromanganese nodules. And just super neat. And the reason to talk about their different formation environments is that it influences their metal composition. And so the metals that are accumulated in these nodules might very well be very different from the metals that are accumulated in manganese nodules out in the abyssal plain, which is where we hear a lot about nodules forming in those environments, and so it's always really neat. Those can have inputs from the sediments. When the redox conditions become more reducing, the metals can be released from the sediments and accumulate in those type of nodules. There can be mixed, where you can get metals from both seawater and the sediments. And it'll be great to check out the chemistry of the nodules collected here to see. But it looks to me like there's a lot of clues that they are likely hydrogenetic, so more similar to the composition of a ferromanganese crust. It's just incredible thinking about what has supplied all of the nuclei that initiate the growth of nodules in this region. I'm curious to find out if any of the volcanologists on the line think that this could be a lot of volcanic debris that, accumulated in this region, and then some sediment buildup and created these perfect formation conditions for the nodules here. It'll be wonderful to see the size of this nodule field if we get out of it on this dive and start to think about the distribution of these nodules on this seamount, to sample them for their chemistry, to think about their formation environment. And then, of course, as we've been zooming in, seeing the different fauna that are using these as a hard substrate for inhabiting this interesting saddle between two peaks on this seamount. Thanks for letting me pipe in.
Thanks, Kira. That is certainly really interesting. I might have some more questions for you in a moment once we take a look at this guy here. This individual.
Interesting.
So, Scott, is this a stock tunicate?
Video?
Yes, exactly. Exactly. Stock tunicate.
Coming down low.
Oh, for some reason the name is escaping me at this moment. But
look at the structure on the inside there.
Yeah, it's very closely related to the vertebrates. It's probably the closest invertebrate related to the vertebrates. And this is another filtering organism, and it's passing water through that body. And you could see on the inside there's some of those structures, they kind of look like netting structures, like basically filters, and they can secrete mucus that cover those filters and then capture small organic particles that are moving through the water column and basically move them along to the mouth that's in there. So a nice flow-through system. You can see the opening on the far side. Water will come in and exit that slit-like opening that we see.
What are the white little balls?
Well, I am not certain if that is storage, byproducts, or if it's actually reproductive material again.
Interesting.
Yep. Yeah, I'd have to go back and look at some of the basic anatomy of this type of
pilot's clear when video is.
I'm thinking it's not the gut. You can see there's something that's kind of brown and winding that looks like a worm in there. I think that's the gut that's leading towards the anus and the sort of upper left inside that chamber. So the next logical thing would be that those are reproductive tissue.
Okay, pushing forward.
Interesting. And you can even see them, and the further we get zoomed out, the more clear you can see them, too. So just before we took a zoom in there, Kira was telling us a little bit more about how these nodules and these types of fields are formed, which is really interesting. So thank you, Kira, for providing that background.
Yeah, absolutely. I'm lucky to be able to join before I go on another expedition. Jason's been willing to share some of the lead geology portion of this dive. Landing on a manganese nodule field in a saddle between two seamounts is not impossible. There's definitely been some nodule deposits or occurrences that have been located on these a little bit shallower depths than the abyssal depths, 5,000, 4,000 meters that we're used to seeing these on sedimented plains in the open ocean between these large edifices. So not impossible, but definitely always a treat to find these and to get to collect them and see what are the different conditions that are forming these nodule fields. Just the
Yeah, we'd like
density
to put
here
in
is
another
amazing.
ship move. We're looking at a range of 30 meters, a bearing of 175 degrees. And
So,
a
Kira, I
speed
think
of
you may have mentioned this a little bit earlier, but for folks who are just joining us, is this something that you would expect seeing sort of in a saddle between two seamounts?
Excuse me, can you zoom on that whip? Thanks.
Yeah,
Yep,
setting up for it now.
They're setting up for it.
There's also a fish in the background or something.
Yeah, expected is a tough one. I don't think expected always, no. It's definitely a surprising find. You can have often, I mean, we dove on a couple seamounts during this expedition, actually. Further out in the cornerized seamounts, we targeted a ridge between two peaks. The name of that seamount we dove on is escaping me. Hopefully I'll remember. And there we just found sediment. It was more of a debris field with some slope failures. And so it's definitely a surprise to find all of these intact, dense nodules here. Like I said, not impossible, but a surprising find.
Thanks, Kara. So it looks like we have a bamboo coral here. And just at the edge here of the shot, you can see one of the nodes.
Let's get it down to the node.
Yes, and excuse me. Yes, this would be either in the presumptive clade Jasonisis, or could be a B clade Keratoisis. We won't be able to tell because it's unbranched. I'm trying to see, maybe Casey, you can see better. I do not see any sharp needles between those tentacles. The tissue itself is quite opaque, so there's probably a lot of scales in there.
Yeah, I'm not seeing any needles right now, but just looking at a section of the branch which is denuded, but with no predator here.
Yep. Not anymore. But, yeah, it's curious what may have formed that in the past. You get a nice view, though, of that protein-constructed node, the dark part, and then the internode, which is deposited calcium carbonate, calcite, actually.
So, Scott, over will the coral sort of regrow tissue over that denuded part if it's left unopposed, or will it always kind of now be in two separate sections for the colony?
Yeah, that's an excellent question, Casey. And our hypothesis is, and this is based on other observations that we have made, is that the coral can repair itself. So it does have the potential to regrow over that exposed skeleton. But it's partly what you were alluding to. There's a competition there. So if other organisms now settle on anemones or crinoids or whatever, that may interfere with that repair process. And so it's a little bit of race to see what's faster, the growth of the tissue or settlement by other organisms on the cleared skeleton. And, this is a little bit of a hypothesis. This is something that we are making conjectures based on all the wonderful observations that we've made on these dives. And so ultimately you would have to monitor that colony for a long term to see how it would respond. There have been some studies along those lines in the Gulf of Mexico, basically following Deepwater Horizon disaster out there. There was a lot of research focus on the benthic deep sea communities and how they would respond to the oil that came through. And so based on some of those studies, we have some good understandings of how these various benthic organisms can respond, recover, and what the time frame for that recovery is.
Thanks, Scott.
We're starting to see more of these larger ferromanganese-coated, I'm not sure we call them nodules at this point for the bigger ones. At least the earlier one we found was very spherical, and the one we collected. So I'm wondering, as we're starting to approach the steeper section, whether we're seeing material that's fallen down from the flanks of the seamount and gotten caught up in this nodule field. And the the ferromanganese crust and the metals have kind of accumulated on them like they do for the smaller nodules themselves. Really interesting bottom type that we're seeing here. Just really, I think we've traversed probably over 150 meters or more since the start of the dive. And we landed in this environment, and we continue to see it. So, and we haven't even gotten to the steep part yet.
I came at a 3.5 right there. Down a couple meters.
Yeah, it definitely seems like there are more boulders in this area, larger rocks, as we're starting to come up. So the purpose of this dive was to start on this flat plane and then kind of work our way up about 200 meters or so during the course of this dive onto the more southerly of the peaks of Gosnold.
Yeah, I don't think I've felt that move yet, but advice.
Copy that.
Still going, or like just stopped very recently?
That was another 30.
That was another 30, yes. Copy that.
It's like
Thank
another
you,
snaffle-branched fish up in the top right, pilot, kind of hiding behind a boulder.
Yep. You want to snap in on the fish video?
Yes, this looks like another snaffle-branched. We've seen a couple of those so far on this dive.
Wait and try. Alright, full wide video. We might be able to get a better view as we get closer.
Hiding away in between those boulders, refusing to come out.
A little bit more tether.
Copy.
Come down another meter too.
Thank you. Looks like it's starting to pick up slope. It's still pretty,
yeah.
Rian, if we're going in that direction, behind the eel, there was one of those small whip-like primnoids where the polyps were extended, and so a view of that would be informative.
Sounds great, Scott. Sounds great, Scott. We're moving in that direction. Yep, towards this cusk oil.
Behind or in front?
Yeah, behind, I believe Scott just said, behind the snaffle-branched there.
Copy that. A little
Yeah,
bit of tether.
it's on the next rock, and I understood the movement and all that, and also for whatever reason I'm pretty delayed today, so I apologize. I'm probably getting you these things late.
That's okay. That's okay. We're heading in that direction anyway.
Alright, we can try and sit up a little far away if we're concerned that the polyps might retract if we...
Yeah, I actually haven't identified it myself yet as to which one we're looking at.
Copy. So this is the little line of rocks that the eel was hiding behind.
Yep.
So if I sit up here, I think I might be able to see it. It's leaning at like a 45-degree angle.
Yeah, there's like two there.
Depending where the whip is, I think it's in front of the fish now.
Okay.
In front of the fish. All right, go ahead and start coming in, video.
Yeah, these little tiny whips are kind of all over the place here.
There's a little white whip. This one here, I'm assuming?
Yeah, actually that one does look like the polyps are out. Yes, there we are. Okay, so this is one with the polyps out, these little primnoid whips. They are so small.
It's quite small. I could attempt to get closer. The last time that happened, the polyps retracted, I believe.
Yeah, thank you for that. That'll be helpful.
Yep, that would be great, pilot.
Earlier, sorry, man. I noted earlier that back in 2005 on Yakutat Seamount, we did see some very small primnoid whips, largely like this. I can't quite tell right now if it's the same, but those would have been in the genus Convexella, species Convexella youngersoni. Thank you for the close-up.
Yeah, we're just undergoing a pilot change here for lunch, so we'll continue on in just a moment.
One. There's also a significant layback.
Copy that. What's the distance?
Approximately 200 meters.
Thank you. Appreciate that.
It's really interesting to see while we're here during this pilot change, little static view, to see the variety of different rock types being coated by ferromanganese. Here in the bottom right of the screen, you can see some smaller material, and then these pretty uniformly sized manganese nodules, and then these larger boulders. Still trying to figure out a good reason for all the different types of rock that have accumulated here, and especially what could be the nuclei for this manganese nodule field that we're seeing. Trying to figure out if it could be a volcanic process. Kevin Conrad in the chat says sometimes you can get explosive volcanic eruptions at this depth, but he can't think of an instance of a basaltic volcano producing this much submarine bombs and blocks, especially on this scale. Maybe Jason can pipe in with some other ideas on where this debris is coming from.
Right by the lasers here?
Yeah, I'm actually thinking if the flanks of these seamounts are
we
largely unstable. They're probably more unstable.
Yes, we are.
During and right after they were constructed, but they do tend to erode, and the flanks do fall down in landslides, just like landslides on land. Landslides occur underwater as well. I tend to see the biggest landslides on the edges of the continents, like along the east coast of the United States, in more heavily sedimented areas. But here the seamounts do erode, and perhaps it was a process where part of the seamount failed and a landslide occurred, and the material was broken up and small enough to form so many nuclei for each of these nodules, but big enough that the current that sweeps through here and keeps the finer sediment from accumulating on top of the nodules. That they were just big enough that current couldn't move them. So it was perhaps the perfect storm, so to speak, of material cascading off the flank of the sediment of the seamount to sort of initiate the process here. We would be able to test that through looking at the sediments and structure below this environment, usually by a long core. Get a big tube full of sediment that gives us the long history of processes such as landslides. But we'll also look for evidence of that as we approach the steeper flank to see if
Go ahead, watch it.
There's a process there left in the rock record that
absolutely. We'll just leave them on.
will help us
No problem.
at least hypothesize some more about it. It's hard to prove any of these things. We usually come away with more questions than answers, but that's always good.
So it looks like contours get closer together up here.
Pilot, this is Watch Lead.
Go ahead, watch.
Can we zoom in on that potential Bersingid left-hand side on the side of a boulder?
Yeah, we can do that.
Thank you.
Come on in, video. Is it this one, watch lead, or the
Yep,
one?
that one there. There's a couple of crinoids in front and then a Bersingid in the back there. So both these crinoids and this Bersingid are both echinoderms, so they're in the same phylum as sea urchins and sea stars. The Bersingid is a type of sea star. They have these really distinct long spines coming out of each arm. And the position that this Bersingid is in right now is kind of a fishing position, so picking particles out of the water column here as they go by. Thank you so much, pilot. That was great.
Yep.
More of these soft corals, too, I can see on the rocks that we zoomed in a little bit on earlier, as well as some polychaetes, some serpulid polychaetes that form these white tubes on these larger boulders and rocks.
Nice slow zoom out there. What were you saying, co-pilot?
Okay, so contours are going to get closer together up here, I guess. So
Copy.
Keep an eye out for an increased rate of slope. But
There's an interesting accumulation of looks like ferromanganese crust debris up against that bigger boulder. We know from
walking across the surface.
Discussions with the pilots and others on the way down that the current through here is pretty strong. So it looks like there is still some movement of finer grained material. And in this case, getting trapped behind the. I'm just going to ignore that biology. So the processes are pretty strong, and those weren't small pieces. So the currents are quite intense through here.
Stop moving as soon as we.
Yeah, so this crab here is a hermit crab. It's Parapagurus, and on its back there has a type of anemone called Epizoanthus, and so it does have a shell. We see these are, relatively common in the northeast Atlantic, and we've seen quite a few today already on this field of these ferromanganese nodules. Thank you so much, pilot.
Pilot's clear. Thank you, video.
Yeah, as we're getting more of these larger boulders, we're starting to see some of the larger fauna too. There's some sponges. I can see some of the anemones that we've zoomed in on previously as well as some larger sponges as well.
So we're stationary here. You're looking down at me. Once I push out a little bit, maybe we can get another move in.
Sounds great. Yeah, it's pretty flat, so.
Copy that.
We could probably cover 30 meters, no problem.
Pushing forward. Tether looks to
Are
be
you
in good
happy
shape.
with 175? The target's at 192, but it's not a hard target.
I think 175 works anywhere.
Yeah.
Anywhere southerly, would be good.
Yeah, I
So
concur.
it's really interesting on that previous zoom, some of the smaller rocks starting to look, these nodules looking more angular, maybe signaling that these are looking a little bit more like coated debris, and we're seeing a lot more bigger coated cobbles and boulders as we approach the slope. So kind of leaving that really rounded, larger nodules that we were seeing previously.
Yeah, stand by, Jim.
So
We'll poke the ground over and get that in.
Think about how this landscape is changing as we move up slope.
I think here's as good a spot as any.
Yeah, Kira, it would be pretty interesting. So the pilots here are going to set up and again kind of move the manipulator around in the seafloor here and see whether it's more sedimented or whether it's remaining in these round nodules. It definitely seems to have changed textures from the very beginning of this dive here.
Yeah, absolutely. And I think Jason hit it on the head when he was talking about the high currents here, and they've been asking, does this seem like it's an expected finding to find nodules here? And I think it seems like
pushing down like 40%.
We could have seen another pavement with these really strong currents here, but a deep enough depth and enough substrate to make a more of a ferromanganese crust pavement. And I think the key ingredient here is the availability of material on top of sediments that is allowing nodules to grow in this region, which is definitely an unexpected setting that we landed here today.
Thank you for ramping up hydraulics.
No problem.
Give it a quick poke here. All right, arm coming on. 3, 2, 1. It's pretty loose there. Watch lead.
Oh yeah, still pretty loose particles there. Yeah, if we can make kind of a mark and then let the sediment settle and have a zoom in on it, that would be great.
Absolutely. Is that big enough mark, or do we need to give it more?
A little bit more would be great.
Copy that.
Just to confirm. Nice.
Drifting just a little bit. Is that mark big enough, or do we need to claw a little more?
No, I think that's good. Interested in the composition of the sediment that's kind of underneath these smaller rocks and nodules here.
Copy that. Stow the arm while the sediment clears.
Yeah, where the skid was too might be good to look.
Oh yeah.
At that to just be a little more broad.
Yeah, that's perfect.
I don't know if you quote that pilot, but they're suggesting looking at the skid mark too.
Oh yeah, that did a better job than the claw did, didn't it?
Yep. But really interesting to see that this bottom is very much more kind of debris-like necessarily than the rounded, very distinctly rounded nodules that we were seeing.
All right, I'm going to set up right here, and I think we can zoom in on that. Stand by.
Yeah, definitely an evolution in the texture from everything being rounded, sub-rounded to now, a more distributed mix of angular, rounded, large, and small. It does kind of lend itself to the idea of some sort of debris. Failure from the flank of the seamount.
Oh, shells.
Yeah, it's really interesting here, Jason. Kind of in the front of the shot here, it looks almost like flakes, like pieces have kind of flaked off.
Yeah, it just beat me to it. So, and I think we were kind of seeing that earlier, and Kira mentioned that we were able to see some of the texture on broken pieces, and it's hard to tell if these are broken or they were, it's the underlying shape of the pieces is broken, and then the ferromanganese coating sort of encrusted them. It's a really, really odd morphology.
Yeah, it's strange enough. I wonder if we should suction sample some of this so that it could be identified later.
Yeah, while we're here, I would be up for that.
Yeah, do you have any targets in particular, or just a little bit of everything with the shells? Yeah. Okay. Yeah.
Some of it looks like it could be shells, and it looks like others in the chat room have mentioned that as well. So I don't know that it is, but it's certainly.
Do we need to reposition it
would
all
be
here?
interesting
to it, or are we okay?
to see
that might be about right.
that and a slope collection. Will hopefully
closer.
A little closer.
Allow that to be identified.
Not much.
Yes, the pilots are just going to set up here to take a section sample of this debris material. So
a couple inches
we'll
forward.
give them a little bit of quiet to concentrate on their collection here.
And back down. Maybe do like, push down like 40, 45, 50%.
Copy
and
that.
enjoy like that in, and then heading. Yeah,
and
that's what I would do.
Okay. Are we going to be able to draw out okay here?
Yeah, if I might have to starboard wing in to avoid that rock, but other than that.
Copy that. Relatively stationary here. Hydraulics still up?
Yeah.
Copy.
There's a sample in one already.
Copy that.
Jar 1.
I think we'll just go to the next course one, since that one doesn't have a filter. We can. Is it 2 that has a course filter?
She suggested a 5, so I'm on 5, but I could certainly do...
5 works. Yeah, we can do 5. Yeah, whatever is easiest.
Yeah, I overheard her say she...
Alright.
Alright, I'm partially extended on the drawer here.
Okay.
I can extend more if you need it.
We can start with that.
Okay.
Yeah, thank you. Arm coming on. 3, 2, 1. I think this should be good enough.
Could wrist left a little bit. See that? Yeah. If your shoulder azimuth support maxes out, I can draw out a little more.
Copy that.
Catch my drift. I think that's a pretty good grab. And then once you get away from the drawer a little bit, then I can draw back in.
Copy that.
All right. I'm going to draw back in?
Stationary.
All right.
Thank you.
And then I'll get that sample box.
Thank you.
Just to clear the view here.
I'm just going to orient the wrist here a little bit so we can see what we're looking at. And we can come on a little bit on the main HD if you can video. Thank you.
All right, so I'm going to draw 5.
Try to get a good look at those shells.
I believe jar 5 is vacant. Nav, is that true? Jar 5 is vacant?
Jar 5 is vacant.
All right, great.
Try to set it down. It looks like we're poking right there where the shells are.
Let me know when you want me to begin suction.
Copy that. I'm going to try to dig in just a little bit here. I think we're go for suction right about here. Does that look okay, watch lead?
Yep, that looks perfect.
Copy that. Let's go ahead and ramp up.
All right, that's 800. Let me know if you want more or less.
Copy. Try to get some. There, we got some of those shells I just saw. I think that's what we wanted. Did we need any of the rocks here? Watch the. Oh, looks like we got some.
Yeah, I think a little bit of everything is good, so we can have another swipe maybe.
Coming back in. Lifting up a little bit. That's a good little patch right here. There we are.
Look at that. It's
Try to give it a little shake. Are we getting anything down the hose? Is that max suction?
I can increase.
You got copy.
There's quite a bit in there, yeah.
Okay.
Let's see. I'm just going to increase suction a little bit to
Yeah.
make sure, because as the filter clogs a little bit, it's going to reduce my flow a little bit. I still see some business coming in.
Yeah, I couldn't tell if the shells
I'm
made
just
it in.
going to want to stop suction and let it kind of fall off the filter just a little bit.
Copy that.
And then I'm going to begin suction again in a second.
That looks good.
And then here we go. So there's quite a bit of chunks in there.
Okay. Does that look sufficient, watch lead, or were you wanting anything else?
Could we do one more swipe? You know, while we're sitting here, we might as well get a good collection.
Sure. Do we have enough suction with that jar engaged, or do we need to move to another?
No, I think that's
okay.
I see how the filter kind of cleared itself.
Yeah, copy that. Do you have any targets in view, watch lead?
Directly underneath where the suction hose is right now, that looks again all that kind of shelly material.
Okay. Is that just to the right of where we collected then?
Yeah. Yep.
Okay. Copy that.
Thank you.
Come on out just a little bit, video, if you could. Thank you.
Okay, I'm going to give you 800 mark.
Copy that. Coming back in. And that looks pretty close to where we want. All right, come on up.
Pretty cloudy in my jar. That's
Copy.
good. It's good.
A little bit more right there, maybe. Get a little scoop in there.
All right.
That looks great, pilot.
Okay, I'm going to stop
Copy
suction
that.
for a second. I'm going to let the jar kind of settle off the filter.
Mixing out. We'll stand by and see what we have in the jar.
I'm going to kind of impulse it.
Nice. Yeah, a lot more debris in there now.
Yeah, quite a bit.
Yeah, that looks like a really good sample right there.
Copy that one.
Okay. All right. I'm going to go to 6, and then I'm going to exhale whatever's left in the tube.
Copy that. I can bring the hose up a little bit. Ready when you are.
All right. I think I'm under a 6. Here we go. Clean that tube out for us.
Oh, we still had some in there. Looks good though.
Okay.
All right.
All right.
Are we? Can we come back to full wide on the Zeus video? Okay. Copy. Thank you.
I'm going to let me know when you want me to extend the drawer.
Copy that. A little sideways there.
All right. I can extend here.
Copy. Stand by.
Okay.
There we are. All right, let's go ahead and extend. It's looking good there.
Nice.
Thank you.
Beautiful.
Thank you.
All right.
Much appreciated.
Awesome. Nice.
That should do it. Thanks for the help.
Yeah. Nice sample.
Why, thank you. Taking off. And if there's nothing else, you can take your camera back too. Yeah. Let's see what angle you're looking down at me at still. I think I need to push out a little bit.
Yeah, I think what's happening is layback is like decreasing just ever so slightly.
Yeah.
Combination of layback decreasing and the last of the ship move kind of making it to the end.
We'll push up a little bit.
We could do a quick snap zoom on this stick sticking up out of the sponge. That would be great as we go by.
Sure. Go ahead and come on in, video. Nice and slow if we could. Thank you.
Yeah, on the top of here we see some large brittle stars on top of this old skeleton. Not quite sure what that skeleton is from. Another one of those primnoid whips and some old sponges at the bottom. Those sponges look like they're just skeletons. That's great. Thank you so much, pilot.
No problem. Come on out, video.
These brittle stars like to get themselves up into the water column to be able to pick particles out of the water, and so they found this old stalk of either it's a coral or maybe a crinoid, just an old stalk that's been left behind to get themselves up into that water column. If you just joined us here today, I want to say welcome to Dive 16 of the NOAA Ocean Exploration Expedition North Atlantic Stepping Stones. And we've been stepping our way back towards the east coast of the U.S. We're in the New England Seamount chain. And today we're exploring a seamount called Gosnold. We're actually exploring a small saddle in between two peaks. We started the dive today at 3,220 meters.
Yeah, same. Yeah.
When we landed on this dive, we actually landed in a manganese nodule field and have been slowly working our way towards a more uphill area. The goal of today's dive is to try and transit a little bit up and see how the different fauna and geology changes through these transition zones.
Bring Mini Zeus back up?
Yeah. Got you.
Thank you very much. Looks great.
It's been a really interesting dive. For many reasons, the geology has been spectacular in terms of seeing a bottom type that we haven't experienced during this dive. We weren't expecting it, based on what we knew about this region, but there's a lot we don't know about this region, and it's kind of, the reason that we're here. We're exploring, we're trying to learn more, fill in our gaps in knowledge. It's a big piece of sargassum.
Yeah, it's a really big piece of sargassum. So we're currently in the Sargasso Sea, so there's sargassum all around the ship right now as we dive the ROV. And as organisms grow on the sargassum, it actually gets heavy and is transported down to the deep ocean, actually becoming a food source for many organisms down here. Thank you so much, pilot.
Come on now.
There's also a couple of sponges on that rock as well.
Thank you, video.
So Jason, we landed in this area where there were really, really distinct nodules, so very, very rounded ferromanganese accretions onto that central core, that nucleus. And now, we just stopped and collected some material from this area as well to kind of compare. But it looks much more broken up. So is this still ferromanganese, or do we think that this is something else?
I think, we're certainly transitioning out of the completely rounded nodules that we saw when we landed. At that point, we're maybe 150, 200 meters away from our current location and away from the steeper part of the seamount that we're slowly approaching. The current conditions and the availability of material from the flanks of the seamount are likely different across those two areas. As we continue to approach the start of the steeper section here, I think we'll be starting to see more of this debris kind of morphology to the rocks. They're still ferromanganese coated. They still have this, the thick color here, the thick coating in the black and browns that we're seeing. But we are certainly transitioning to a much more complicated bottom type with different size material and different shapes. And the reason for that last suction sample was we're seeing things that kind of look like shells. They were not what we'd expect from rock debris or from ferromanganese crust sort of chips that might come off. So it's kind of interesting to make these leaps in the bottom and try to understand how the topography or the bathymetry of the seafloor is affecting or being affected by the currents and what we see when we get here.
Yeah, these are
lots of processes at work.
Yeah, these are a little unusual sponges. I don't think we've seen anything like this quite so far during our expedition. Scott France says these are potentially bolosoma. They look almost like little mushrooms on the seafloor with these stalks and the caps on top. And these sponges are filter feeders, and you can really see the pores, the circles in the top cap there. Beautiful. Thank you so much, pilots.
You're welcome. Thank you, video.
We can see more of these small primnoid whips, and actually you can get a good good idea of the size of these primnoid whips that we're seeing. Those lasers that you see, those two red dots, those are two harmless scaling lasers, and they are 10 centimeters apart.
Pilot,
and
the direction
they're used to
175.
help us scale organisms and seafloor features as we're diving today.
Go ahead, watch it. All these little ones.
Try to find a larger rock with the one.
Yeah, copy that. Copilot suggested we might be able to find one on a rock somewhere. It'd be a little easier to access.
Like those two little guys?
Yeah.
On the right, bigger rock.
Oh yeah, I see that.
Maybe put it in a buy box.
Do we need to halt the ship move, or do you think we have time?
The ship move should be just about done.
Okay.
I can ask.
Copy that.
Yeah, you can stop the ship, I guess. That's fine.
Copy, Bridge. Thank you.
So you think we can grab these and
Yeah,
put them in?
I think so.
Copy that.
Make sure that's the right species on there.
Yeah, we'll take a quick look at them.
So on the seafloor here, ever since the beginning of the dive, we've seen these small little primnoid corals. They kind of see them sticking up. They're like little, almost like little feathers out of the seafloor. And these are a little bit unusual and very, very characteristic of this area. So we're going to attempt to try and collect one. It's just finding the right one to be able to pick up because they are very, very small.
I don't know if that's the right one on the rock.
Yeah, it may not be.
This one has a little anemone on the rock there as well.
Yeah, and this you can grab that small bit.
Right.
If it doesn't crumble.
Is this the species that you're looking for, Watchlead, or a different one?
Yes, yep. So it's that the long, thin coral. You can see the polyps sticking out on the side there.
Oh, on the little rock.
Yep,
Yeah.
on the little rock with the anemone at the base. I think they're both on the same rock there.
Yeah, copy that. Do we just want to grab the rock or just snip the...
If we can grab the rock, that would be perfect.
Okay.
It's going to be quite small.
Yeah, we'll see what we can do. Alright, come on out, video. Go a little bit closer here. And we have plenty of time, Copilot?
Yeah, I think it'll be okay.
Copy.
I'll let you know.
Yeah, please keep an eye if you don't mind.
Absolutely.
So this should be accessible.
Looks perfect.
Looks nice and stable.
I concur.
Copy that. So should we get the claw next to it? I don't think we're going to probably be able to get the rock, yeah?
You could potentially
so.
kind of get the jaws in the sand and then carefully grab that little rock, but have to see what that rock looks like next to the jaws. What are you thinking?
Yeah, we can probably put the jaw next to it and go from there.
Once you turn hydraulics on, then you can do grip force.
That's right.
So hit exit. Okay,
All right.
now turn on.
Are we ready and ramped up?
Yeah.
Copy that. Coming online in 3, 2, 1.
And then grip force will be available to you.
Yeah, I might want to go in with a slightly lower grip force.
Yeah.
Okay. Can we come on in just a little bit partial video? Thank you. That's good for now. Thanks.
What bio boxes are open?
The only box we have anything in is starboard forward.
Okay, great.
Alright. Come out just a little bit video, please. That's good, thank you. Just wanted to see size of this relative to the claw. It looks pretty small. That might be difficult to pick up.
Yeah, maybe we can find a bigger rock. Or this larger one by your right toe.
Is that? Oh yeah, that could potentially be
the easy one to snag.
Yeah, let me just index out real quick. Watch the rock to the right on the right toe of the ROV. Do you see that? Is that the right species as well?
Yeah, if we could have a quick zoom in to confirm, but yes, I think that's the right species.
That.
And that actually might even have a sponge on the front, which would be
I'm just gonna move
a good collection as well.
We're thinking that one might be easier to grab the rock.
Yeah, for sure.
I'm just gonna mosey on to the right here. I think we're going to have a little sit down here.
I've prepped the port bio box.
Copy that. Alright, we can come on in video. We'll just identify this real quick. Make sure it's the right one. Oh, right there on the right. Yeah.
Yep, that's the right one.
Copy that.
Thank you.
Come on out video. Thank you. We just wanted an ID on it. Yeah, that's perfect. Excellent. Is that full wide video? Thank you. All right. You said port bio box is what we're going?
Yeah, this is to port side.
Copy that. Coming on. Oh, I cut it off. Sorry
there it
about
went.
That.
I don't know if it's going to come with it at this point. Might be worth trying to find a different one.
So sorry about that. Yeah.
Yeah, it's going to come right off.
It's trying to. Go ahead, video. Oh yeah, I don't know if that's on a rock that we can actually grab the.
This one down on the front, right?
We could.
Yeah.
Are these too delicate to suction sample, watch lead?
I think we could suction them on kind of a gentle suction. If if the rock is small enough that it would come right through.
If that's an option, we can certainly give that a try, or we can hunt for another rock for you.
Yeah. Let's give that a go, and we should be able to see inside the suction canister as to whether it's survived.
Copy that.
Okay, so what are you thinking?
Yeah, let's. Are we too close to it to draw out a little bit and get a good angle on it? Or, yeah, I probably need to move back just a little bit, actually. Let me just scoot back a little bit.
You could suction the one that actually got cut off of the rock.
Yeah, I was thinking we would try that one and maybe get a bonus on the right there. That should be enough room there. All right. Yeah, I think we can draw out a little bit here. Thank you. Coming back on.
You can draw out further if you want.
Yeah, that's perfect. Thank you.
Which jar should we do? Nav?
Why not 2?
Copy that.
Just pull out a little bit here. Alright, go for drawer close.
Okay.
Thank you. Alright, and then try to get a good angle on it. And if we could come in just a little bit video, that'd be great.
Got mini zoos too.
Thank you. That's great video, thank you.
Let me know what you want. I'm going to start at maybe 300 or something.
Yeah, 300 would be great to start at.
It's 300.
Try not to get too much sediment along with it. Oop. Stand by. Doing some roll of the ship here. Alright, let's try again. I think 300 might be enough.
Okay. I'll maintain.
Go ahead.
That's 300 still.
That's kind of buried in there now. We can increase to maybe 400.
Okay, that's 400.
Try to rock a little side to side here. I think we're pushing up against that rock there. There it goes.
Alright, keep an eye on that jar. I'm going to maintain this level of suction.
Copy.
Seemed like it was moving at a good speed. I might have to increase a little bit.
Yeah.
But keep an eye on that jar and let me know.
Copy that.
Could increase a little bit, I guess.
Yeah, we'll have to see.
Oh,
there it
is.
goes.
All right,
I see it
stopped
confirmed
the
in there.
suction.
Thank
I'm gonna
you.
Go to 6.
Yep.
All right, and then we can. I want to draw you out.
Copy.
A little bit, and then we'll keep going. Sirius moved a little bit, so.
Copy that. Yeah, we need to unfortunately probably move a little bit. Watch lead. But
all right,
if we need to go for another collection, we can at some point.
I actually think that's great. It looked in good condition inside the canister there. So,
Copy that.
yep.
Thanks for your patience.
I'm gonna blow to clean the
No, thank
tube
you for
a
that
little
collection. That was a hard one.
bit. It was definitely in the jar, right? Yeah.
Yeah, it's in there.
Okay. Awesome. Okay. Suction hose is clean for the next sample. And there we go.
Stand by. There we are.
Okay.
Go for retraction.
All right. Nice.
Appreciate it.
Bring a mini Zeus up
Copy.
for blue view ops.
I think that's the. Can't tell if the shoulder's all the way up.
Yeah, looks fine.
Copy.
Okay. So I'm set.
Hydraulics gear.
Okay. I'm gonna.
Before we leave this location, pilot, there's a slightly different coral, kind of directly behind the larger sponge, slightly right of center. It's kind of tucked in behind it. You can just kind of see these branches coming out.
Copy that. Stand by. Do we have time, co-pilot?
Yeah,
Thank you. Just a quick snap zoom is great.
Is it right behind the sponge here?
It's so look up a bit further.
That guy right
That one right there.
Copy. Go
Thank
for zoom.
you.
Quick zoom video. Thank you.
This is a bit of a larger form of coral. And another bamboo, I think. Oh, the polyps are in, but this looks a little bamboo-like.
Absolutely, Rian.
Yeah, unfortunately, all those polyps are tucked in. And it's out. Yeah, there we go.
They are tucked in. I actually don't see any nodes right now. There's one.
There is one. Yep, I was just about to say I saw one.
The shape of the polyp suggests maybe actinomyces.
Yeah.
The tissue is so transparent.
Very.
I'm probably close to the end of the move.
Very, very transparent. And the tentacles are very well retracted in there.
Yep. Yeah, so these look like the little volcano-shaped polyps that characterizes actinomyces. So this could be a young one. If it is, this would be the deepest known record of such a thing. So I'm not saying with any certainty that's what it is.
Thank you so much, pilots. That's great.
No problem. Copy. Thanks, video.
Yeah, I think
Keep our eyes open for that one.
Just a little bit.
Copy that.
The layback's
I'll push
actually
out.
increasing.
Oh, it's
The
increasing.
current's gone up.
Okay.
I will push out. See you looking at me at about 160. Push starboard as well. Heading 175.
Yeah, my heading is being blown off just a little bit from its set point.
Copy
But
that. Just get a little in front of you here, and then we can explore.
Sounds good. Blue looks pretty clear.
Hey, Ria and Kira here. Still hanging on the line, enjoying these views that we're going over this incredible debris field coated in ferromanganese, and in many cases we've been identifying this widespread deposit as manganese nodules when we first landed here. Lots of very rounded, very obviously manganese nodule deposits growing in concentric layers around nuclei. And now as we move more towards the slope of the feature, we're approaching getting some more angular debris coated in ferromanganese. When you stop calling it a nodule is a bit of a difficult distinction. Typically you use that for rounded concretions that grow in these concentric layers as they precipitate from seawater and sometimes from fluids that are being released from the sediments.
Go ahead,
but we can definitely still see this very uniform debris field coated in ferromanganese. And we're trying to figure out what could have provided all of the nuclear material for this
zoom on that boulder
deposit.
video.
Is it volcanic debris? Is it material that's fallen down slope? And this really high energy current environment keeping this region totally free sedimentation, and so allowing a lot of this ferromanganese to precipitate and create these nodule deposits and coated debris fields, and some of these larger rocks looking like coated cobbles, maybe large pieces of debris. This one coated in ferromanganese, but also this interesting white.
Yeah, I just, we've seen a couple of these ripples, and we weren't sure if they were worm tubes or sponges, but I think we can confirm they're actually sponges forming these funny lines. When I first saw it, I thought it was a different color on the rock, but it's actually a sponge kind of coating the edge of this. Thank you so much, pilot.
No problem. Pilot's clear. Thank you.
Yeah, it's really interesting to have landed in that nodule field. That wasn't at all what we were expecting on the dive this morning. And so far we've seen some really interesting biology, mostly small forms. So where those nodules are very mobile, if you were there at the beginning of the dive, you will have seen the pilots gave it a poke and kind of moved it around, and it was very, very easy to move. And where you get that movable bottom, you don't tend to get these larger forms. But now we're, as Kira was saying, we kind of moved into this area where we're seeing more of these larger boulders. And so we're starting to see a little bit larger in terms of the fauna down here.
What is the current layback right now, Nav?
There's a little tumbleweed of sargassum going by on the bottom left. It's the sargassum is up in the upper water column. We've seen a few actual large pieces so far on this dive.
It's a lot of layback.
And it's an algae, a photosynthetic algae that lives in the upper water column. But as animals grow on it, it becomes heavy and heavy.
Same as last.
And then eventually drops down to the seafloor.
That works. Thanks, Nav.
Tumbleweeding its way across this bottom here.
Bridge ROV nav.
You're welcome.
I'd like to request to move range 30 meters.
I feel like we're seeing a little bit more in the way of kind of larger patches of sediment in between some of these kind of this debris. Seeing more of that glinting. It kind of glints out white, cream color. And when we first landed, there was just none. It was a field, a mass field of these ferromanganese nodules. And here in the distance, you can kind of...
I would guess that as we have...
Oh, go ahead, Kara.
Sorry.
Nope, go ahead.
Oh, I was just going to briefly add on to what you're saying that, as we move towards this slope, we're probably seeing some more downslope material leaving those areas of exposed sediments. It's just amazing. It's so fun on these discovery-based dives to see what we find down here. We might very well have also seen a ferromanganese pavement coating rocks, because they're seeing enough sediment accumulation in the past. It's created this environment where these coated debris and nodules can form. Just so fun to see this as we dove between the saddle between these two peaks. Not impossible to find, but definitely exciting to find this type of morphology here.
Trying to decide if the current shifted the other direction or not. Or if we just turned around a little bit.
Yeah, the tether's not doing anything too crazy at the moment.
Yeah, this is looking really quite different now. A lot more in the way of kind of larger, I don't know, are they called boulders when they're that size? They're still not terribly big, less than 10 centimeters across, but definitely seeing more of them. Oh, pilot, on the left-hand side here, I think is another one of these bamboo corals.
Copy.
And this might potentially be a target for a collection. This one's a bit unusual and has the scientists in the chat room here a little stumped.
Copy. Set down for a zoom here. Ship move currently underway now.
Yes, it is.
Copy that. We can go for a quick zoom here, video. Is this the one, watch lead?
Yep, that's the one. We're going to take a look at it. Scott France is on the line. I'd appreciate his thoughts, but this looks pretty similar.
Do you want me to stop the
I
move?
am still here on the line.
Yeah, if they're going to go for a collection, perhaps we should.
It's, it's certainly, this is the bamboo coral we were looking at earlier. It's got that really thin skeleton in there and very transparent.
Could we come to an easy stop,
please?
please?
You can see the polyps are extended this time, not contracted.
Thank you, Bridge.
I can see on one of them sticking out to the left, you can see there's some bright white lines between the tentacles. So I'm presuming those are the needles. I can't quite see the needles, but I presume that's what they are. Intertentacular sclerites that help us narrow this down. I don't know, Rian. It may be that it's too small, and that's why I can't really distinguish it. It's got interesting shape to the skeleton. You can see that it's really wavy. It's not growing very straight.
Thank you, Bridge.
Interesting angles.
It is interesting because I usually think of the bamboos as having really quite straight skeletons. They'll branch off, periodically, but those branches tend to be very, very straight. So this one's definitely a little bit different. And it's really curious to see two small ones.
Yep, and it's almost like a well-organized bramble type,
Right.
Exactly that, but that's the closest I can think of in terms of this very irregular branching. Very irregular.
Copy that. Yep. Clear video. Come on out.
Can you tell which rock it's attached to?
It's hard to say, isn't it? That
pretty
little one almost
large
looks
polyps
like it's
relative
the big
to
one,
the size
maybe if we get Lindsay's on it.
Yeah. I'm stumped.
Come on out, video.
Yeah, I think we're going to actually collect a little piece of this one, Scott, if we can, because this one is definitely a bit unusual.
Yeah, I can't even see where I couldn't see nodes clearly enough to know anything about the branching. I suspect it's internodal branching, which would go along with what I said earlier, that maybe this is
what do you think as far as
a very young colony of an ectomyces. But as I said, this is extraordinarily deep for what we know about
to
that
the right
genus.
here.
I don't think that genus we know from deeper than 2,000 meters.
For that.
At 3,200, that would be very unusual.
Not at the moment.
You could try.
Yeah, it'll be very exciting to
parking on top of that.
Yeah, it'll be very exciting to see this collected. And so this is a good time to remind everyone that collections from this research expedition and everything from the Okeanos Explorer actually goes to a national repository.
Yeah.
And so when we make collections, we preserve them carefully, and they go to a national repository where any scientist from around the world is able to collect those and take a look at those specimens.
Got enough reach here, you think?
Maybe not. Try turning to port a little bit. Get closer.
Kind of nestled up on that little boulder there.
Got it.
Feeling some tug or some current, I can't tell. May just be having a hard time sitting on that little boulder.
Yeah.
Yeah, because I don't have a skid down. I think it's just bouncing on that boulder.
Okay.
But we might be able to get stable here. Give me just a second.
Could move that rock too with the manipulator.
Yeah, I might need to. Yeah, I don't know if we can get close from this angle. Watch lead with our current tether. There's a little boulder in front of us, so we might need to move first.
Okay.
This guy right here is making it difficult to land.
Oh, I'm not sure if that one will move, but...
Yeah, it's quite large.
No.
What do you think, co-pilot? I could maybe...
Yeah, you can see where your skids need to be.
Yeah, let me try to position myself with that again one more time.
Yeah, you can probably move it.
With the manipulator?
Yeah. Yeah.
Okay.
Just bring it off to starboard.
Copy that.
And then park right there.
Let's give it a shot. Yeah, I think we can handle this one. Watch lead. Give us just a second.
Great, thank you.
Mm-hmm.
Hydraulics are up.
Copy. Thank you.
Yeah, and then you can just swing it starboard.
3, 2, 1. Copy that. Armor's live.
Or closer to the sample.
Yeah.
Whatever.
Somewhere. Give us more room.
Pilot, watch lead.
Go ahead, watch lead.
Is there any chance of putting that in the rock box? Or is it too big?
What do you think, co-pilot? Is that beyond our maximum size?
No, I think you could do it.
Yeah? Yeah, we can collect this watch lead.
All right, let's do that.
Would we like it in the port or starboard rock
There's
box?
something in starboard.
Okay, in starboard or?
Starboard forward has something in it.
Okay.
A geological sample.
Copy that, so we'll need to open the drawer. Stand by. I'm going to set this down.
The port forward's vacant, is that correct?
According to my documentation, it is correct.
Awesome. Alright, I brought this port wing inboard so you can open the box.
Thank you. Much appreciated. Now we can collect it, watch lead.
Perfect, thank you so much.
No problem.
Pretty high up.
Copy.
Watch this camera.
Yep.
You can close the drawer
from
the
top,
top too, yeah, whatever.
I like to look at it this way, but I will try that next time.
Dealer's choice. Nice. Awesome.
Almost.
Alright.
That works. Thank you.
Port forward.
Just going to keep the arm out over here.
Sounds good.
And we will move ever so closer here. How is Sirius settling out?
Good. You're off to my port side, but you're still in view, and I think I'm probably pretty much done moving.
Copy that. I'm going to try to avoid the sponge off to our port there a little bit.
Okay.
You think that's enough reach?
Yeah. I'm curious to see where he's attached.
Give that just a second to settle. Yeah, it's hard to tell, isn't it?
Yeah. Maybe Mini Zeus might give us the answer.
Yeah, we'll definitely zoom in. Do you want to zoom in with Mini Zeus first before I stick the arm in your way? Or should we go?
At the same time, it's fine.
Okay. Coming on, 3, 2, 1. Put the claw next to it. And yeah, if we can come in on Mini Zeus video, that'd be great.
Video, do you need more light on the mains? We got enough? Okay.
It's quite small.
Yeah, I'm curious what it's attached to. Looks like it's attached to that kind of nodular one right there, right?
The larger one?
Yeah, the one.
Yeah, it might be.
That's what it looks like. Yeah, I'll give you some more. Here you go.
You can come in on main Zeus as well, video, a little bit.
Yeah, I'm thinking that one.
Yeah?
Right?
I could try to move the rock and see.
Yeah, like give the rock a nudge.
Oh, yeah, I think you're right. I see it attached right there, yeah.
See the circular?
Yep, from this angle.
And if you carefully...
Put that in the bio box?
Yeah.
Okay. Yeah, I think we can do that.
Okay. Retracting drawer. Retracting pins. We'll go for port. Okay.
Let me get a good angle that will not disturb it. Go for a lighter grip force here too.
Just kind of pluck it out of there. Hopefully it's not a large...
Very nice.
Got it in my fingertips. Let's see if it stays.
Okay. I'll follow you with Mini Zeus.
Copy.
You want me to draw out?
Yeah, please come out there.
Alright. Port side should be ready to go.
Copy. Do we need to zoom on this, or are we okay?
No, I think let's stash that so it's safe. Thank you.
Yep. All right, we go on port inner or outer?
Whatever you want. Dealer's choice. Does look a little precarious. I would probably go inner.
Inner.
In case it crumbles or drops.
Definitely have more room in the outer, but just don't want to damage it when it goes into the box.
Yeah, you go for outer. Whatever.
Yeah, let's go for outer if we can.
Sounds good. Nice. Alright. You want me to retract?
Let me make sure. Is the claw out? Yep. Go for it.
Alright.
Thank you.
Nice.
Nicely done, pilots. Thank you so much for those 2 collections.
You're very welcome.
Nice. Alright. Looks great. I turned to port a little bit to keep you in view. Just keep that in mind. If you come starboard with me.
Taking off.
So I was just informed that the USBL on Sirius isn't tracking properly. The readings on D2 should be correct, but you can't trust the ones on Sirius.
Okay.
It's going in and out.
Okay. Alright. Let's get our heading back southbound.
Copy that.
So I'm turning to starboard with you here as you come over.
Sounds good. I'll lateral starboard. Oh, we got a fish ahead of us.
Ooh.
Go in our general direction. We might be able to get a zoom on him.
Here's a fish.
Okay, I'm lined back up.
Fortunately facing the other direction as always.
Swim away, swim away.
Yeah, won't be able to get a good look at him, unfortunately.
Yeah, unfortunately I don't think he's facing the right direction for an ID, sadly.
Alright, we can come on out, video.
Okay, I'm lined back up, kind of the 175 marker, you know, pretty close to 180. My set point is at least. I think we can get another move in. Unless there's something exciting right here.
Is 175 still the angle, or do we want to turn a little bit more westerly?
Yeah, I don't know if waypoint 2 is a hard target. I think we
No, the targets aren't hard targets.
Okay, just uphill.
And we don't have any...
okay.
Just follow the interesting terrain.
Okay.
Yeah, sounds good. 175 works.
Bridge ROV nav. I'd like to request a move. Range 30 meters, bearing 175 degrees, speed decimal 2
So if
knots.
you're just joining us today, I'd like to say welcome to Dive 16 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones.
Good copy, thank you.
And we've been stepping our way back into the east coast of the U.S. along the New England Seamounts. We started at the Corner Rise at the beginning of this expedition and been working our way west. And so today's stepping stone that we're examining is Gosnold Seamount, and we're actually in a little saddle area in between 2 peaks of Gosnold. And we started this dive today at 3,220 meters.
Bring hydraulics down.
Copy, thank you.
So we started today's dive in a really interesting area, landed in a manganese nodule field, which was very, very unexpected for this particular area and depth. And we kind of came into an area of more rubble, but it seems like the bottom here has got more rounded again, more nodule-like, along with these larger boulders that are in the middle. Seen some interesting biology so far as well, mostly small forms. You know, these manganese nodules and kind of this rubble area is very mobile bottom, so only smaller organisms tend to inhabit those areas. When we looked really closely into those nodules, we saw things like isopods and brittle stars, and then some of these smaller whip corals that you can just see. We've had a couple of collections already today of a couple of different coral species, and then several of these rock types to try and identify what kind of nodule they are and how that they were formed.
we, we're definitely seeing, and it has been noted in the chat that, the bottom seems to have this kind of ripply, undulated nature to it. It's not, when we first landed, the pavement was very, I won't say flat, but it was, it didn't have sort of undulations in it. It was all kind of at the same level. We're moving, there's a big, actually big coral skeleton there. So I think we're really moving into an area where debris is coming down off the flank of the seamount, and it's starting to accumulate, and we're starting to see, a lot of different sizes of material. We're not seeing the sort of same size nodules as we saw earlier. So, keep saying as we get closer to this wall, things are likely to change, and it does seem that way. Where the current is actually pushing material around, and we may be seeing these undulations may actually be representative of the debris apron or the big pile that forms at the bottom of the slope if material is falling down from above. So, it's a very interesting bottom substrate morphology that we've been going along. Diagnostic of a number of different processes, but still lacking any of that fine sediment at the surface. Speaking to the strength of the current through the area.
Yes, for sure. And one of the things that we noticed this morning as we came onto this dive site is that the current on the surface is quite high, but then also the current as we were approaching the bottom was also quite high as well. This is definitely a very dynamic area, kind of in between these two peaks, where potentially the water is squeezed a little in between those peaks and goes a little bit faster than it would in the surrounding areas. On these larger boulders, we can see these white sponges kind of encrusting on the edges here, and sponges are filter feeders, and so they like to be in areas with at least a little bit of current, but sometimes really quite high currents. They're picking particles out of the water column, picking nutrients out of the water column as the water moves through them. Definitely seeing some of those stripes, Jason, potentially those ripples in the seafloor here.
Yeah, they're really starting to appear, and normally when we see those type of features, it's a current flow generated feature where there is a certain level of sorting of the different size material. And that's certainly possible here. The current is actually really quite strong, and it can move larger material. But whether it's strong enough to move some of this really big material, I'm not sure. That's an interesting sponge.
Yeah, it could be it.
Yeah, all encrusted in lines, and then one up on the stalk as well.
Coming back towards you. Okay.
Yep.
It's pretty fascinating. So, it's really not as clear as normally would be in a more sedimented area that it's the current pushing everything around to form these ripples, or it's it's reflecting a different process. But every time we move, there's another interesting thing here to look at. And, it's been an amazing dive already.
It really has, and something very, very different for this expedition as well. We certainly have not come across one of these ferromanganese nodule fields before, or any of this kind of habitat. It's been very, very different, and quite the surprise find this morning. Pilot, this is Watchley.
Go ahead, Watchley.
Could we zoom in on the sea star?
Yeah,
It's a
absolutely.
Brisingid.
Slowly making my way here. Thank you, video. Still moving in on it, so stand by.
This is a Brisingid sea star. They're kind of instantly recognizable by these long spines on their arms. And this one has its arms up into the water current and is kind of fishing for food. These Brisingids pick particles out of the water column. And so they use those long arms to catch particles and then bring them forward into their mouths. And the mouths are actually underneath what we're seeing right now. We're kind of seeing the back, the top side of the sea star here. Thank you so much, Pilot. See a crinoid on the rock there as well, some forams, potentially some bryozoans. You know, every time we zoom in, we see a lot of life around the bottom. Christopher Moss says this particular Brisingid might be a Freyella, and that's certainly a name that we've recognized from several of our dives so far on this expedition. These deep water Brisingids. We're certainly seeing a lot of the sargassum on the seafloor here as well, which is interesting that the sargassum is landing in this particular spot. We've seen a few pieces in previous dives, but nothing quite like the density of it that we've seen so far here. And interesting, the bottom here, you know, on the right-hand side, we could see those really distinct rounded nodules. And then here, it's kind of more of this debris that we'd zoomed in a little bit on earlier.
Sirius tracking in probably 15 minutes.
A bit of a change.
Yeah, Nav made us aware of that a little bit ago.
We snap zoom on this sponge in the
foreground.
foreground.
Yeah, it really got more finer in terms of we're not seeing so many more of those nodular features anymore. It's more of this. Almost piles of just rubble.
For sure, yeah. You can
little bits and pieces of cobbles and boulders here and there.
Yeah, yeah. You can really see in the background there those pieces. This is a beautiful sponge here too, and there were some of the soft corals, and in the background you can see those little primnoid whip corals that we had seen earlier, and actually managed to collect one of as well. There's a beautiful crinoid on top of the sponge, and you can really see the texture of the sponge, kind of this knitting together of these spicules. So these sponges are made out of spicules. This one is probably silica, hexactinellid, and those all knit together to form the structure. Really beautiful. Thank you so much, Pilot. That's great.
No problem.
Brittle star on there as well. You can see several little polychaetes. The sponge is what we call one of those ecosystem engineers, so building kind of the structure that other organisms like to live around on the seafloor. And you can really see it in this picture. Lots of organisms kind of living around this sponge and using it for habitat.
Nice job. Copy, thanks.
Several people in the chat room here are saying that this sponge is potentially a conolasma. See a couple of these here, and we've seen some as we've moved up to the spot as well. So this research cruise is brought to you by telepresence. So telepresence not only allows you to be able to see the seafloor in your homes today, but also by scientists from around the world. And in the background here, you'll hear us talk about scientists in the chat room. And there are scientists from all over the globe that are helping to identify organisms and seafloor features today and on each of these dives. And we really appreciate their expertise in helping us to identify what we're seeing.
Same as last?
I think so. How do you feel about a move, co-pilot?
Well, we just lost the Sirius Center.
Yeah, how do we want to... we did that... I think we lost that in the shakedown as well on one of the dives.
But just looks like it's coming back.
It's intermittent.
Yeah, I mean, if you come up and we do like a little scan, but what are you at, 60 meters? It's like maybe something. No, I don't really see anything out there.
It's been pretty flat mostly.
Yeah, we could put another move in.
30 meters, 175.
That works for me.
I think that works.
There's an interesting conversation in the chat room right now about these small little primnoid whip corals that we're seeing. Every time we get close to the seafloor, you can see these little short little whip corals, not more than 10 centimeters above the seafloor, and just how mobile the seafloor is. You know, we're not seeing a lot of those big forms that we've seen on some of the previous dives of this expedition, where we're seeing corals that are several meters high. And one of the reasons for that is the floor here is a little bit mobile. You know, when we put the manipulator arm into the seafloor a couple of times so far on this dive, it's moved around. It's very, very mobile.
Just put in another move.
And so it really can't sustain those large corals. So, but we are seeing these small little primnoid corals, which suggests that it's not extremely mobile. It's not moving on a regular basis to allow these smaller, shorter corals to be able to live and grow there. There's also potential that these particular primnoids are maybe a little bit faster growing than some of the larger forms that we see, and so are able to grow in shorter timescales in between the movements of the rocks that they are sitting on. Oh, pilot watch lead?
Go ahead.
So there's a coral right in the bottom center frame. It's just going out of frame now. Could we have a quick zoom on that? I think that's either an Okeanos or a Chrysogorgid.
Bottom center here?
We haven't seen that so far today.
We have plenty of time here.
Thank you.
No problem. Is it the one right under the lasers here?
Right to the right of the lasers there.
Right, yep. Gotcha. I see it. Go ahead and come on into you.
Very hidden.
Yeah, it blends in. Nice spot.
It's very fine bottle brush coral here. Yeah, I think this is a Chrysogorgid. We'll wait for Scott to chime in.
I will chime in that I concur with that that guesstimate.
Very large polyps.
Yeah, large polyps, relatively large polyps on a very fine skeleton, very, very thin tissue. You can kind of see underlying that the skeleton itself has a sort of goldish color. The Chrysogorgiidae translates as golden coral. And one of the keys about the Chrysogorgia is the very regular geometric pattern to the branching. So much so that one of the characteristics that used for describing the species and identifying the species is to count the number of revolutions of the main stem as you're going upwards to where a branch comes to overlie a branch below it, and whether it's rotating to the right or left. So when you read species descriptions of this, you see 3L2, 3 turns to the left would give you 2 branches, that kind of thing.
Oh, interesting.
General Chrysogorgiids are very regular geometrically.
Ah.
2 little there.
There's some pretty big amphipods in here, too.
Just gonna check your blue view out.
Copy that.
That's really great. Thank you, pilot.
No problem. Pilot's clear. Take your time, video.
Now the seafloor here, we're starting to see those regular nodules again. So we're out kind of on the rubble and more into the regular rounded nodule shape again that we really started this this dive in. So that's really interesting as well.
Yeah, it certainly seems to be more patchy in terms of, you know, those rounded boulders, and then you get into that finer stuff in between, which is probably the more angular material that we did get some collections of. So we're jumping from patch to patch, from nodules to other things, and you know, as we move up slope, I think we'll start to to see more in place or larger material that may reflect another one of these transition zones.
Still underway, correct?
That's correct.
We keep saying we're going to get to the slope. and we're slowly going up it. It's just not as abrupt as some of the ones that we've been going up on previous dives. So
yeah, we should definitely say the slope is not as dramatic. Definitely not as dramatic as some of the slopes that we have been on, or even the steep cliffs that we've been on during this expedition so far. This one is definitely more of a gentle Sunday drive on this Friday.
Looks like a change in the sediment up ahead slightly, perhaps.
Yeah, I think up ahead we're seeing kind of more of these boulders again. So kind of in this coarser grained area in front, and then we're moving up to maybe some small boulders. So maybe we're going to hit that transition zone that you were mentioning there, Jason.
Yeah, no, I think certainly you should start here, and keep my eye on on the Blue View sonar. Folks at home can see that on the Stream 3. There's a quad screen display of a bunch of different tools that are being used by the pilots and Rian and myself and others to get a sense of the environment around us beyond just the visual. And the Blue View, which is in the bottom right-hand corner of that feed, sort of is a sonar that's allowing the pilots to see up ahead, see what's coming, so they can make decisions about driving the vehicles, and we can use it to sort of get a sense of what might be up ahead and what we can expect to see. So, and also on that quad view, the top right is the HIPAC screen, which is a navigation screen showing the position of and the ROV and Sirius, all on plotted on top of the symmetry.
Thank you, Bridge.
Of the region, and then the pilot GUI, so you can see a lot of the information that the pilots are using to fly the vehicle.
Which one?
Over the bottom.
Bright yellow, okay.
Well, both vehicles.
Come on in.
And then there's the view from Sirius, which you can see on Stream 2 in bigger format. So
this is interesting.
Certainly a lot of stuff coming up.
Yeah, and this is interesting. So at the very, very beginning of this dive, we saw one of these yellow crinoids with some sargassum wrapped around it. And here we are with another of these stalked yellow crinoids with this sargassum around it, which is really interesting because I wonder, how they shed this sargassum. So these crinoids are, they're suspension feeders, so they need to be able to pick particles out of the water. And here it is wrapped up in the sargassum. So is the sargassum actually going to kill this organism because it's not able to feed anymore, or will it eventually work its way loose? But this is the second of these stalked yellow crinoids that we've seen wrapped up in a big clog of sargassum. Really interesting. I think at the very bottom of this rock in front too, Pilot, there was potentially a sea star. I don't know if we can pan down a little bit.
Copy that. Coming down.
See more of these soft corals. Brittle star, I think. A little brittle star
okay.
Wrapped around.
Come on in, video. Nice and stable here. Might as well get a shot.
Yeah, this one's definitely tied itself up in knots. You can see the beautiful spines on this brittle star. And I think it's kind of sitting either, so there's a little old fossilized coral stalk that's sticking out from this rock, and the brittle star is actually wrapped around it. You can just kind of see it poking out towards us. Beautiful. See some bryozoans. And then more of these things that look a little bit like shells, but we think are maybe kind of flakes off of the rock down below. We actually had a collection of those earlier, so we'll hopefully be able to identify them when they're brought up to the surface at the end of this dive. Though
if we're still here, can we get just a quick look on the top of the rock back where the crinoid was, just to its right.
To the right of the crinoid on top of the rock. Yep.
Yeah.
It's,
There's some yellow net-like structures. Just want to verify that that's what we're seeing. So maybe a bryozoan that we saw on an earlier expedition a couple years ago.
Kind of yellowy color.
Determine what it was.
Come on in.
Yeah, exactly.
Yep, I think it's to the right of the lasers pilot. It's kind of on the edge of the rock. Like right underneath where the sargassum. That's it.
This little guy here.
That one right there.
Gotcha.
Those are small.
Yeah, I think actually that one might be a hydroid. I can see the polyps on that one.
I agree with you.
Yep.
It's not growing where I thought it was. Really nice coprella to amphipods below it, the skeleton shrimp.
Oh, yes, yeah, the white skeleton shrimp. Yep.
Yep.
Doing okay
and
here,
then another one,
yeah,
this is the third or fourth of those really spiky sponges that we've seen in the background, which I suspect is the cladorizid. Thank you for the close-up. That's good. That's definitely new for the day.
Thank you so much, pilots.
Absolutely. Stand by, watch lead. We're just going to do the next one.
That zoom provided a really good view of that material that we did collect some of, and hopefully we'll be able to identify it. But it sure looked like those were shells, ferromanganese coated shell material. It rather than being little pieces of rock. So that's really interesting in terms of. We haven't seen a lot of large shell debris on these dives, so where is it coming from, and what does it mean in terms of this sort of nodule field?
Yeah, it's really interesting.
A lot of unanswered questions so far.
Definitely a lot of unanswered questions, and I'm glad that we managed to collect some of that rubble earlier, that hopefully we can identify whether it is shell pieces or something else. And in amongst those, there were pieces of urchin test as well. So these hard urchins, when they die, their kind of hard outer shell breaks up, and you often find that in rubble piles on the deep sea floor, and there was definitely some of that in there. So it's really be really intriguing when we open up those suction canisters and find what's there.
Okay.
It's always a little bit exciting when D2 comes back to the surface with all its loaded with prizes, and we get to go and unload the D2 baskets and have a look what the collections were for the day.
That's certainly the fun part.
Look. Looks like we got Roland over there now. How you doing, Roland? Oh, okay. That move completed a while ago. Is that correct?
6 minutes.
Oh, 6 minutes. Okay.
Pilot, this is Watch Lead.
Go ahead.
Would it be possible to just have a zoom in kind of on the rubble, the finer rubble?
Yeah.
I feel like there's now a lot more white in there than we've seen earlier. Like it's changed, the composition's changed a little bit.
Yeah, you come in on the here.
Just as we're going by here. Yeah, this looks quite different, Jason. So a lot more, yeah, definitely more of that kind of rubble, potentially shells. I actually wonder if they're barnacle plates, looking at them. Sometimes these long gooseneck barnacles leave behind these large plates. See other shells in there as well?
Yeah, those are definitely shells.
Yeah. One of those small little primnoid corals there. Some bryozoans.
Even little gastropods and
yeah.
It's a whole mix of
Across.
And this looks almost more recent, like there's more, there is, obviously those black ferromanganese coated shells in there too, but also a lot more white shells, which suggests that they haven't been there certainly as long as the black ones, but maybe not terribly long.
Yeah, there is this, kind of down slope, and I think, most of this material is obviously coming from something on the steeper section above us. That's the good thing. It can't come from below us, so it can. We sort of have a good idea of where it's coming from. So it suggests that there is, quite a, either a fossil accumulation of these fauna. We've seen, with the cup corals, we've seen this big, on a lot of our dives, these big debris piles of fossil cup corals, and I'm really wondering, where these are coming from and how they.
I'm okay with it when pilot's ready.
What that community and habitat looks like, because we haven't really seen this before. But
Bridge ROV nav.
As we get closer to the wall, a lot of this
I'd like to request to move.
Newer material gets trapped here first before it gets moved down slope later.
175 degrees, speed decimal 2 knots.
That's probably why we're seeing, this more new or fresher or uncoated material, rather than what we saw downslope, which was all pretty heavily encrusted with this ferromanganese.
Thinking we should be close a little bit
oxide.
More rise of the slope here.
We're not done with the interesting parts of this dive yet.
I think the whole dive is going to be interesting, Jason. It's definitely, it's been absolutely fascinating so far. So we're diving kind of in the saddle area between the two peaks of Gosnold Seamount. And the reason that this dive site was chosen by the community was we're really interested in this depth. We started this dive at 2,220-ish meters, and this is a very understudied depth in the New England Seamount area. But the other was to have a look at this feature kind of in between these two peaks, and this is an area that we see in other places that really funnels water, so you get some high currents, and that's certainly what we experienced on the way down. And so it would just be a little bit different from diving on an area which was, maybe a ridge arm or one of these other features coming off of the seamount. Being able to dive in this kind of unique V-shaped feature is really interesting. And it's certainly proven that today. We're quite deep, so we're not going to come across a wall feature, but certainly up above us, further up the line, if we were able to dive, continue diving D2 for several days, we'd eventually come up to much shallower depths and maybe hit some wall features on the way up. And so a lot of this material, as Jason was saying, is potentially transported down from much, much shallower depths further up this feature and transported all the way down here.
Yeah, co-pilot, how's layback looking?
Uncertain, since we don't get any reading from
we're still not getting anything, huh?
D2. Well, it just changed, but I'm not sure how accurate it is.
Copy that. What's the distance to D2?
To D2 is about 180 meters.
Okay, we can kind of approximate it from that.
Nice.
Very interesting.
It does seem like a lot of this, shell material type stuff is getting trapped behind these larger rocks, and even this small distance we've gone where, the color of the bottom has changed dramatically from that kind of consistent, black, dark brown color to, a lot more of the white tan of this material that just isn't yet coated with ferromanganese. some crinoids.
Yeah, certainly a lot of crinoids that we've seen so far. Some of them stalked, and many of them sitting on these larger rocks, and also on some of the sponges that we've seen as well. These crinoids are filter feeders. They like to be up in the water column a little bit, or certainly able to stick their arms up in the water column.
Maybe you can frame this one up.
It's a beautiful sponge here at the base of this rock.
So get some more tether.
Even from this distance, you can really see those pores. So these sponges are filter feeders. They have all these holes, these pores going through their tissue, and inside those holes are specialized little cells called choanocytes, and they have little wiggling flagella, and these little wiggling flagella create a water current that passes water through, and then there are other cells in there as well that pick out all the nutrients and really good particles for these sponges to eat and feed and live. And right on the top there are two white-yellow crinoids kind of blended in really well on the sponge. These crinoids are often found on sponges because these are really good areas to get that water flow and to catch those particles as they go by. Beautiful.
I like how it's formed around those rocks perfectly.
Yeah, the bottom edge pilot is all kind of in and out of these rocks, and the sponge is kind of grown around these rocks, and very often sponges will actually grow over features as well, so who knows, maybe there's a few of these nodules at the base of the sponge. Really beautiful. Megan Putt says this sponge might be an Atlanticella sponge.
When was that last move complete? I'm trying to figure out if we should call another one.
It hasn't completed yet.
Okay. Video, you want to snap the in the water column here while we wait? Looks like this is the shrimp or something.
A hard one to focus on. That one was quick moving.
Yeah, I lost it. Interesting grouping of rocks there.
Yeah, these kind of larger boulders that are here.
Thank you, Bridge.
So does that still count as a botryoidal texture, Jason, when they're that large? These are pretty big.
Do you want to keep moving? Or?
Perhaps if they are not cemented together.
Wait a second, yeah.
Although, those ones do look, almost look like a stack of cannonballs sitting on the seafloor. And, over time, some of them may actually get cemented together by, subsequent precipitation of ferromanganese crust. So, we see a lot of loose ones here.
Looks like quite a bit more of this type of terrain for a bit.
These ones have been here longer and, over time ended up getting cemented together. We're seeing, just behind that one is this elongated, boulder, perhaps outcroppy type piece.
Did you want to snap this?
We're definitely moving, into an area where we're not seeing everything being, spherical. There are more shapes coming. Again, pointing to that change that's coming ahead in terms of the slope. We might start seeing more in situ or in place parts of the seamount that may be the underlying volcanic, substrate. That thing is moving.
Yeah, this Parapagoras is not very interested in being photographed. Thank you very much. This is a type of hermit crab in the deep sea, and it has either a epizoanthus or an anemone on the back. And we've seen quite a lot of these so far.
Thanks, video. Looks like you're starting to feel that move.
Yeah, I think I'm creeping forward a bit.
Video, and it snaps in the white circle on this rock.
And actually, Carol Mizell mentioned earlier that, on previous dives, although these are mostly loose, and we've been collecting a lot of these loose nodules on other dives, we may have seen these, but more as a pavement where all the nodules may have been cemented together, probably by ferromanganese crust.
Excitement.
And I think we're starting to see more of that process here.
Just a tad.
These larger boulders. So there's this kind of continuum of different processes that we're seeing.
Another one of these.
I'm not sure that's a sponge, I believe, or an anemone. With a couple of crinoids.
I keep working my way up.
Sounds good.
Yeah, that was quite an unusual looking anemone.
I'll work over to the stock sponge.
Copy.
Then we could probably get another one in.
Yeah, that's what I was thinking.
Okay.
30?
Yeah, let's do it.
Not too much biologically diverse this dive is, but I'm seeing that there's quite a bit of sponges and possibly tunicates, as well as there was, I think, a cup coral seen earlier.
I'd like
some
to
occasional
request
fish.
to move range 30 meters.
A few of those crabs that were seen
175
before.
degrees, speed decimal 2 knots.
You can zoom in video, partial.
Kim, I think perhaps it is pretty biologically diverse. It's just that most of the fauna that we're seeing is really small, and so we're passing over it without even realizing how much is there.
Absolutely, you're right. I mean, part of what's on this hard substrate are a lot of these hydroids and different things that sometimes you just can't really see unless you're zoomed in all at once. This looks like an interesting sponge.
Lots of different sponges, and even I think we've seen 4 or 5 different coral species today. I think this is a sponge in the genus Caulophacus, which would be a
Zoom out to
rustellid
get the full
glass
specimen
sponge.
video. So I'm curious how its stock shape is interesting to me.
Yeah, and it looked like there was a brittle star on the top there, but I can't tell if it is still alive, that kind of grayish one, that really small right on the bulb area.
Yeah, I agree. There were definitely some live ones on the main stalk. I'm not sure if that upper one was alive.
Stock is pretty interesting. It's like
Oh, pilot, can we get a zoom right on the left-hand side? That was just about to come out of screen, but it looks slightly feathery, almost translucent.
Yep. Okay, video come in. Small, translucent.
Were you looking at that barnacle, the yellowish one?
Come in video.
No, it's coming into dead center now, being pulled in. I'm not sure what this is.
Okay. Well, after we look at this, which maybe is a bryozoan, but after we look at this, we should get a look at the what I think is a barnacle, because Jason and Rian were earlier talking about how possibly what we're seeing are a lot of barnacle plates, so there's a live barnacle. I think this is a hydrozoan.
Okay. Yeah, it looks quite complex and very delicate.
Is that max zoom video? Okay.
Great, thank you, pilot.
Yeah, video can come out.
Scott, where did you happen to see that?
Just bump it up. It's going to be basically right above this hydrozoan, a little bit further on, and you'll see it's yellowish.
Oh.
There we go. It just came in the top of the screen.
All right, I think it's going for the
All
back
right.
here. You want to snap
I
in
think
video?
dead center now, pilots.
That guy?
Thank you.
Yep. I can get closer. I'm pretty far away from it if you need a better shot.
Should we get closer, Scott, or is this a good enough view?
It's a good enough view just to demonstrate that these barnacles are down here. They're alive. These are crustaceans, and right now it's closed up, so you don't see its feeding limbs, but it's those plates that are at the top that Jason and Rian were hypothesizing that may be a lot of what we're seeing here that are forming this platey encrusted material, which would suggest that at one time there were a lot of these barnacles.
Got a little tether now.
That's what those are.
Looking down 5-3 degrees.
Yeah, it's kind of hard to visualize, whether or not it was, just a higher abundance all at one time, or if we're really seeing a longer-term, process of these barnacles living down here, and the geologic record is long, and accumulation can be kind of slow, and we can preserve really long records. We've talked about the Desmophyllum cup corals and how some of the skeletons that we find on the seafloor of the fossil ones can be 15, 20,000 or more, up to 100,000 years old. So we may be seeing a really long sort of time period of growth and death of these barnacles, and whether there was a field of them all alive at one time, and the water conditions just changed, some environmental criteria that was no longer suitable, and they died off, or we're actually seeing the more common sort of abundance of them, and it's just a really long time of accumulation, and the lack of sediment accumulation from the water column, and driven by these stronger currents in this area, may just be leaving these larger materials that can't be moved by the currents to accumulate.
Jason,
so it's
the other part I think that you're basically alluding to there is the fact that they become encrusted with this presumably ferromanganese crust also preserves those shells for a very long period of time. If they didn't get encrusted, they would probably degrade faster and we wouldn't see them.
So Scott, while you're on here, take a look at this.
Yes, I think this is a benthocodon. So this is a jellyfish that likes to spend its time sitting on rocks. Maybe it wants to evolve into an anemone or a coral one day. Who knows? But beautiful specimen. I think Dougal Lindsay will be really happy when he reviews the tape to see this.
Absolutely gorgeous.
I said hydrocodon. I think it's benthocodon.
Benthocodon, yep. Yeah, so are these actually attached to the rock itself, Scott, or do they literally just sit here like this?
My understanding is they're literally just sitting there. I don't know if it's, part of the day it sits on the bottom, but we certainly see these swimming in the water column. And it's possible, if we disturbed it, it would do that. But it's not a permanent resident of the substrate.
Yeah, it's absolutely stunning, beautiful. Thank you so much, pilot.
Yeah, pilot's clear.
Good spot.
You can come out, yeah. All right.
I should point out, I believe benthocodon is a hydromedusa, so it is a jellyfish, but it is within the group Hydrozoa, which is different from the lion's mane jellyfish that maybe you're familiar with in shallow water, which would be part of the Scyphozoa, the true jellyfish. And so the hydrozoans have this alternation where as one stage of their life, they may be small polyps, and then they have another stage of their life where they're medusae.
Yeah, I got scared. That was the one I was going for. All right, come in video. I must have touched the ground, and then he got scared. All right,
that was
come
awesome.
in on this one.
That was really cool. Yeah, so I think that was a Cereantha that just went shooting up into its tube here. And in the front here is an Anthomastus, which is nice to see. So Anthomastus is a type of soft coral, otherwise known as a mushroom coral, and has these big, long polyps, but actually has other polyps as well that are much smaller and tinier, and you can barely see them on kind of the mushroom cap of these particular corals.
Next video.
Beautiful.
See if there's other subjects on this rock of interest.
You can just see the tube left behind by that Cereantha that sucked right up there. Oh, actually, the pink on the right side of the rock would be really interesting to take a quick look at. That might be something a little different.
I'll get you closer video here. Okay, come in video. Yeah, you can take it. Not a super good angle on it, but
Now this is interesting. This looks a little bit Paragorgia-like.
I agree with you, Rian. It's most certainly Scleraxonian, which is a larger group that includes the family Paragorgiidae.
You're in good shape right now.
Okay.
Yeah, quite a small little colony here.
Is that a picnic?
Sorry.
No, go ahead.
Sea spider just in front of it.
Oh yeah, the pycnogonid. Yeah, I was just looking at the snails right above it. There's actually quite a lot of things on this side of the rock. You know,
A lot.
Yeah. You can see.
Sponges and soft corals. And
yeah, on this.
I was going to say that for that Scleraxonian, that Paragorgia-like thing, it has an interesting skeleton that's quite different from a lot of the other octocorals in that it's more like concrete cemented together sclerites with live tissue passing through it.
Yeah.
Thanks, video.
Yeah, so lots of things on the edge there. Pycnogonid sea spiders and the stoloniferous octocorals, so the octocorals that grow kind of single polyps growing off of a stolon or a kind of thin ribbon, and then that Paragorgia. So definitely starting to see some different organisms as we move along our track here.
Onward and upward. Ooh, what is this red subject here? Gonna snap it video? This might be
Nav. It is 1840.
Looks like one of those gelatinous sea stars.
Yeah, so this is one of those slime stars, I think.
Slime, is it?
Actually, moving along this rock.
Got more zoom video, or is that
yeah.
it? Oh, he's got more. Oh, sorry. Flip. 1 second video. Sorry.
Came up a tad, looking at you about 60 degrees.
So these, we think this is potentially a slime star, which one of the main features is they have this kind of outer covering on the outside of their skin. So the skin is actually has kind of the second covering and a hole in the middle, and that hole allows water in and out.
Okay, pilot's clear when video's clear.
Christopher Ma, our sea star expert on the scientific chat room here, says it's potentially a pteraster, certainly an odd one, potentially a Corinthastrid. I think I totally butchered that one. Sorry, Chris.
Corinthastrid. You guys have very difficult names. Corinthastrid. All right, headed uphill, co-pilot.
Copy, looking good.
If you're just joining us here today, I want to say welcome to Dive 16 of the NOAA Ocean Exploration Expedition North Atlantic Stepping Stones. And we are currently on the New England Seamounts and moving our way west towards the east coast of the USA. and we're exploring a stepping stone called Gosnold Seamount today. And the dive today started at 3,220 meters. And we have been diving in kind of a saddle feature in between two peaks of this particular seamount, and I've definitely seen some very unusual things today. We have about 40 minutes left of this dive, so I wonder what else we can see during this dive. Pilot, this is Watch Sleep.
Go ahead.
Can we actually zoom in on the sediment kind of under this underhang here?
Incredibly reflective. I'm getting the flare off it.
Very reflective.
Come in.
He beat me to it.
There you go. I'm starting to read your mind, Jason. We've been doing 16 of these dives together. So.
Yeah, it's getting kind of scary.
Yeah, some pteropod shells.
And Ian, I'll just note, as you look at this, as you were zooming in, I saw there were at least 3 barnacles on the rock above here, so if you're looking for a live barnacle.
We should definitely look at that when we finished here. Thank you, Scott.
How am I doing, co-pilot?
Doing okay, back at about 60 degrees on you. And I'm centered at 175-ish.
Should be right.
There we are. So barnacles. So
a lot of them.
Yeah. So we see some of these plates that are covered over in this ferromanganese patina that looked potentially a little bit like barnacle plates. We've actually have a collection of them on board D2 right now, and so when D2 is recovered and back on the Okeanos Explorer, we can take a look and see what those are. But we've been wondering whether they're actually the plates of these barnacles. You can see these barnacles have these plates around them, and maybe as those barnacles have died and the shells have persisted and got covered over in that ferromanganese patina.
Yeah, that almost like ridges on them is very diagnostic, and a number of the zooms we've done looking at what we think of those ferromanganese coated barnacle plates, very much have that texture and morphology to them as well. So hopefully, once the samples are back on board, we'll see that and get a confirmation.
Here's one of these yellow stalked crinoids, and is actually, I think, is one of the first, if not the first one that we've seen today that doesn't have a big sargassum piece around the top of it. We've seen a lot of these kind of tumbleweeds and pieces of sargassum today.
Did you need a closer
Interestingly,
look
we're
at
starting to see more of this sort of tabular, blocky material, more angular than that we've been seeing as these kind of cemented nodules. Where, I think we're actually starting to see, more perhaps material that's been displaced from the flanks of the seamount. Still a lot of these cemented nodules and other things around, but well, this may actually just be a cross of those cemented nodules, actually. Now that we have a closer look at it. So that's really interesting.
Is that
almost layered.
Is that the current from D2 pilot that's kicking up?
I would say that's
okay.
I'd say it's super light stuff being thrown up on my thrusters. Like, it was really far in front of me, so it must have been extremely light.
Very fine.
Yeah.
Yep. So if you can hear those alarms going off in the background here, we are just having a practice fire alarm. It's just a drill. So the rest of the ship right now will be going to their stations, and this is something that we practice every week.
Ooh, I think I see a fish up there.
Looks like a slightly steeper slope in front of you.
Yep.
You are out in front of me, though, and I think I'm slowing finally, so...
Copy.
Once you're comfortable, we can go down there, move in.
What am I looking at? pink in color.
Oh, yeah. Can we zoom in on that?
Is it terrestrial?
Very bright pink. Pinky red.
Video, you want to zoom in on the possible strap?
Yeah, looks like a strap or a line.
Huh.
Looks like there might be some rust on it. Maybe a little metal strip of some kind.
Interesting.
Hard to say.
Yeah, a ribbon of some kind on the seafloor here that's bright pink.
Okay, you come out. Thanks, video.
Thank you, pilots.
Yeah. See squat lobster?
Yeah, I thought it's maybe attached to something.
Scott Francis pointing out that's the second time today we've seen unusual color on the seafloor, and it turns out to be debris from above. This really highlights the connection of the shallow oceans with the deep oceans. Anything up in the shallow ocean definitely has the potential to be transported down here to the deep ocean. All parts of the ocean are connected. If you're hearing those alarms in the background over our headsets, they are just drills that are going on right now.
I'm going to zoom in on this. I'm going to actually get a better view of this video. Hold on. I'm going to spin to the starboard. Get like a profile view. You can zoom out a little bit.
These beautiful hexactinellid sponges. This one looks like it has a little friend inside it.
We can let it go, video. I'll keep looking for the items. I'll call workshop. Maybe he's there with headphones or something.
Yeah, I'll double check.
You can zoom out video. I'll keep moving.
Workshop, co-pilot.
You can turn off your XRV.
So Kevin Conrad in the scientific chat room here is saying that this bottom really seems to be that mix of nodules like we were seeing earlier, but also some of these angular basalt debris, these larger pieces as well.
Yeah, certainly at some point we expected to start seeing the volcanic material, and it's having spent so much of the dive in looking at these nodule forms, a changeover to pillow type structures or these more inflated lava morphologies. It's hard to tell when you kind of changed out of one into the other. So it's certainly very, again, just a completely dynamic bottom in this area. A lot to figure out. We're getting a fantastic view of the bottom here from the D2 and Sirius cameras, but it's still only a very small portion of the area. We may cover 500 or 600 meters off the bottom, during a dive, and then go up 200 or 300 meters up the side of one of these seamounts. But it's an unbelievably small fraction of the seamount that we're exploring, and Is it a giant sponge?
Yeah, I was just wondering if that was anthropogenic or a sponge there. Looking up a quick snap zoom on that on Pilot as we go by.
Take a video.
Thank you.
It's probably good.
Yeah, it's actually a sponge. You can see that really tight-knit skeleton, kind of these fibers running through it. So this must have been quite a large sponge that has died and left behind its skeleton. And you can see there's all kinds of other organisms on this as well. I can see some snails and some crinoids using the skeleton of the sponge. That must have been quite spectacular when that was alive. Thank you so much, pilots.
Yeah, no problem.
And another kind of one of those tumbleweeds of sargassum that we've been seeing on the bottom kind of throughout this dive.
I wonder if the Marianas Trench is full of sargassum.
I'm sure it has some.
Could we zoom in on the rock that's just above center right now? It's 3 white, I assume sponges sticking out from the top there.
Stand by video, I'll get closer.
Thank you. This definitely looks like a very different bottom type from what we landed in, potentially less mobile. We might start seeing some bigger organisms as we're making our way up here. Our depth right now is 3,190 meters, so we're still at quite a deep depth and a very unexplored depth for this this area in the New England Seamounts.
3 little guys. Curious what they are.
Actually, barnacles. There we go. 3 barnacles. That I wasn't expecting. So part of the rubble around here we think that we might be seeing is potentially barnacle plates. So these barnacles are crustaceans. So they're actually in the same phylum as crabs and those hermit crabs that we've seen wandering around. And the barnacles actually attach to the seafloor head first. So they stick their head to the floor and then they grow up backwards. And inside those barnacle plates, none of them are out right now. They're little feeding legs that come out the back.
Okay. Come out when you're done video.
Nice. Thank you so much, Pilot.
Just want to do 180.
Again, covered in many of those smaller soft corals and these primnoid whips that we're seeing throughout this dive so far.
Bridge ROV nav. Is now a convenient time to request a move.
Or these sponge skeletons that are left behind on the seafloor. So sponges, when they're alive, can be white or different colors. They can be reds and yellows. And when they die, they kind of leave behind this brown skeleton. And that brown skeleton still forms structure on the seafloor. And so a lot of organisms can still use these the sponge structure once the actual organism has died.
I just scanned on your blue view, didn't see too much out ahead.
See some more sloped crinoids here.
A little steeper, yeah.
And more of these Bursinges and sea star and brittle stars kind of draping themselves over the rocks.
Take a zoom of this Brazingid real quick. For some tether. Come in video on the Brazingid.
Looks like a brittle star hanging out on this rock.
Oh, I lost one.
And again, every time that we zoom in here, we see these soft corals. There's kind of one either side of this brittle star. We can see sponges on here as well, and some foraminifera. And this brittle star seems to be missing an arm. So this particular one should have 5 arms. You can see one is missing. They will actually grow back. You can actually see on the right-hand side, the tip of that particular arm that's really long is very white and not quite the same as the rest. And so it must have been cropped off, and that one has grown back. And so this really short one at the very top of this brittle star has been cropped off quite recently and has not yet started to grow back.
Next video.
Thank you so much.
No problem.
And if you're hearing the whistles and alarms in the background, those are just a drill today. So every week aboard the Okeanos Explorer, we have drills to help us practice what to do in the event of an emergency. And so the rest of the ship outside of the control room today is practicing their drills.
Yeah, that's pretty interesting.
This looks like an old stalk of a sponge, but it'd be great to get a quick zoom in on this. I think these are more of these barnacles that we've seen quite a few of today.
Copy that. Come in video.
Yep, more of these barnacles and another brittle star and a crinoid. All on this this looks like a sponge stalk. You can kind of see how it's knitted together. Beautiful, thank you.
Yep.
I think I saw a large whip coral in the background here too. I think it's still a little bit of ways, but
I'll head there next.
Coming up on that.
Eyes on. Got enough time to make it over there? Maybe, maybe not.
Looking at the surface of these rocks too, I think I see a lot of these barnacles. So that might explain why we're seeing so many of these potential shells or barnacle plates on the seafloor further down. It'll be very interesting when those collections come back aboard the Okeanos Explorer this afternoon. We'll be able to see if we can identify those as barnacle plates. See this large potential whip bamboo coral. We did see one earlier today. I don't know if this is the same kind. Again, as we've moved out of that more mobile, smaller nodules and into kind of the larger boulder field, potentially seeing more larger forms. We're certainly seeing some bigger sponges, and now we're potentially seeing some bigger corals as well.
I am standing by with bated breath.
There you go. Oh, it's all the polyps are all closed up, Scott.
Indeed they are. Again, they look kind of volcano-shaped, so I think you're right. I think this is maybe one that we were looking at earlier today.
I was just to see if there's any that were open. Hopefully not.
Well, that time I suggested it was B1 or J1. I'm actually looking at this.
Could we go down to the base, please, pilot?
Yep.
Thank you. Oh, it has a branch off of it. Look at that.
Yeah. It's very odd the way the polyps are arranged. I mean, it could be an actinomyces. They have the volcano-shaped polyps. And the fact that it's branching certainly would help with that identification.
Interesting.
Sometimes the close-ups just confuse me more than help.
Yeah, it's always the way. Thank you so much, pilot. That's great. Yeah, traditionally a lot of these corals have been identified solely from the skeleton, and that was really just a factor of collecting in the deep ocean before modern technology. We used to only be able to collect in the deep ocean using grabs or small trawls or dredges. And now, of course, we have these wonderful tools like the D2 ROV system, where we can actually go down and visualize the organisms living in their natural habitat and look at species association. And also for some of these corals, it now gets confusing because many of them have been identified on their skeletons. And now, of course, we can see all the soft parts and start to use some of those as potential taxonomic features. beautiful sponge here with some large brittle stars on top. If you can have a snap zoom, that would be great, Pilot.
Yeah,
Thank you.
you can take it.
Beautiful structure on this sponge. You can really see those pores and how these brittle stars are using the top of the sponge as a kind of feeding platform, getting them up into the water column, and they're sticking their arms out into the water current, looking for larger particles to be able to eat.
Like a sword bite, kind of.
Beautiful. Tangled fishing lines there, the tangled arms of these brittle stars. Thank you so much, pilots. That's great.
Okay. Video, you can come out when you're ready.
And again, the background, every time I feel like we've zoomed in today, we've seen more of these little primnoid whips. You know, the density of those particular whips are really quite high in this dive. It's kind of surprising to see so many living here. They almost carpet the sea floor. It seems like every other rock has one attached to it. And that's been the same throughout this dive, even landing in the nodule field that we were in earlier. Can we have a zoom in just on the substrate, just where the lasers are right now? I'm intrigued if all these white pieces are actually barnacles.
Yeah, snap it with the barnacles. Hold that.
These are actually pteropod shells, larger pteropods. So there you go, I was fooled. They're not actually barnacles. It's pieces of pteropod shell which shine back very bright white. Thank you, pilot. That was great.
We have been seeing more of the pteropod shells as we move up the slope here. When we first started, we didn't really see much. And just recently we saw that really sort of large accumulation of pteropod shells of multiple different species. So they are starting to show up a little more, you know, perhaps we're moving out of some of the high current areas. So, you know, different forms of sediment are able to accumulate. Whereas down the base of the slope, the currents were just too high.
Go ahead.
I'd like to request to move, range 15 meters,
We're still
bearing
above
180
the
degrees, bearing
condensation depth.
speed decimal 2 knots.
You know, carbonate undergoes dissolution in the deep sea, so it's probably not a function of that.
Hydraulics up.
And above the aragonite saturation depth, which you know, pteropods are made of aragonite, the more unstable form of carbonate. So, a new story emerging of the sedimentation.
So the pilots here are just going to give the ground here a little bit of a poke to see whether it's cemented or loose. Having stopped here at this location, Jason, this really looks a lot like the nodules that we were seeing earlier, just almost bigger ones than we were seeing at the beginning. Oh no, a little loose. Yep, that moved.
Yeah, so we were really wondering if they were starting to see all kind of cemented forms of these as we moved up, but yep, everything at least here looks loose.
Yeah, interesting. Kind of the larger pieces are more cemented.
Are you okay?
Sorry, go ahead, Jason.
Yeah, you know, questions on, you know, how cemented they are, and, you know, are we seeing, you know, a mix of sort of volcanic lava textures with the ferromanganese textures, and you know, it really is, you know, I think continuing to see some of these, you know, these loose nodule forms, whether they are, you know, entirely ferromanganese or the nucleus on the inside is larger. we obviously won't be able to tell that just from visually. Hopefully some of the samples we collected will give us a picture of that. But yeah, really interesting how much just we can learn from a poke of the manipulator. So that was awesome.
Yeah, really, really interesting. It kind of reminds me of our dive the other day that was deep, and we landed on an area that we wondered if it was nodules initially. And when we sat down and poked it, it didn't move. It was all very cemented pavement. And yet here today we're getting those looser nodules.
Okay. Taking off. Now that I made a nice dust cloud.
Fine sediment being kicked up.
This is quite the drill today.
Yeah, I think they said that.
Yeah, if you can hear the alarms and whistles in the background, they're doing quite an extensive drill today, so they're just having a drill. So outside of the control room, everyone is practicing what to do in the event of a fire or an evacuation. And we do this every single week, and it's to help keep us safe so that we know what to do in the event of an emergency.
Is it 15 minutes left of bottom time now? Is that correct?
13 and a half
13
now.
and a half. Copy that.
I'm going to secure your hydraulics.
So if you're just joining us here today, we have a little less than 15 minutes left of this dive. We're on dive 16 of the NOAA Ocean Exploration Expedition North Atlantic Stepping Stones. And today we've been exploring a deep area on Gosnold Seamount, an area in a saddle, kind of in between the two major peaks of Gosnold. And we found some really fascinating things today. We've landed in kind of a nodule field and have been working our way up slope just a little bit. And in areas that are a bit more cemented and areas that are still a bit looser as we sat down and poked at the sediment just now. Hey, there's two of these. We've seen these. So these are Parapagoras, a type of hermit crab that has either an anemone or epizoanthus on its back. And we've seen quite a few of these today, actually, really quite a lot.
They're two next to each other. I was hoping to get interaction, but...
Hiding behind the sponge. Go ahead, Scott, sorry.
Yeah, no problem. I just wanted to follow up on what you were saying about, what we're seeing today and how interesting it all is. Another thing that's interesting is that wasn't too many days ago we did another dive at around 3,300 meters, 3,400 meters, and we were all remarking on how surprised we were at the diversity that we were seeing, the particular corals and stuff. It sort of exceeded our expectations of that.
Take off.
And today we're at a similarly deep depth, seeing stuff that's surprising us both geologically and biologically, dominated by lots of small things biologically, but very dense. But it's an entirely different community than what we saw at the last site. So just another indication of how much exploration we have to do to better understand why we see the communities we do and the places that we do, and what are all the associated factors that are controlling that.
Yeah, absolutely, Scott. These depths are really underexplored, particularly in this region.
Next video. Wonder if our watch change is gonna get.
Yeah, we might have to do the setup.
Yeah. Video. Let's snap zoom this. I'm just curious what this is. I think it's just a dead sponge, but yeah. Okay.
Yeah, this looks a little like a old sponge skeleton left behind. We've seen a few of these now, and it's absolutely covered in these soft corals as well. I see some forams and some crinoids, some soft corals, lots of life on these sponges.
Thanks, video. Keep going uphill.
They definitely seem to be the larger organism in this area. We're seeing lots of these smaller little primnoid whips, and we've seen a couple of larger bamboo corals, but primarily it's been, for the larger fauna, it's been these big sponges, potentially a coenlasma sponge.
When is that last move complete?
Lots of crinoids on our journey today as well. Okay. Seeing some stalked crinoids as well as some
time for one more.
Crinoids that are sitting on these nodules, these larger boulders.
15?
Yeah.
And some on the edges of sponges as well.
Thank you, sir.
Just going to take a peek at this sponge, I think.
Bridge ROV nav.
Ooh, it's a white one, isn't it? Okay, come to partial video on this white sponge. Just getting settled in here. Okay.
Bridge ROV nav. I'd like to request a move, range 15 meters, bearing 180 degrees, speed .2 knots.
See these beautiful brittle stars kind of on the edge of this this sponge here. You can really see the texture in that sponge.
Good
so those
copy,
pores that allow water to run through, so they can extract nutrients out of the water. See some crinoids on the top there as well as brittle stars, creating a little bit of habitat in the deep ocean here.
How are we looking, co-pilot?
Beautiful.
You're looking good, yeah. Okay. You got a little tether.
Thank you so much, pilot.
Yep. All right, video can come out. We'll keep searching.
We'll just let this last move settle out, and that should be it. Looks like you have a few targets in the area.
Yep. Quite a bit of those hermits here, huh?
Yeah, another one of these hermit crabs, the Parapagoras, hiding under the ledge there. And we've seen more than a dozen ashore of those Parapagorids, these hermit crabs with either an anemone or Epizoanthus sitting on top. That's kind of a little sediment swale here up in the foreground, up in the background. Interesting little area here where sediment is accumulated. Those finer particles have found a place to settle. And what kind of looks like all these big nodules all kind of joined together. I don't know whether that's pillows at that point, or are they still nodules when they're that large?
I think it's something we've been going back and forth about in the chat room is what these morphologies are reflecting, and certainly that poke that we just had with the manipulator suggests that these may still be just nodular forms, whether they're all ferromanganese or just very thick crust. They are cemented together. So some are loose and some are cemented, and the cemented ones are forming these more tabular blocks. But little sponge. Oh, and an octocoral. Not sure what that is. That another coral there.
Sorry, the alarms are going off behind me there, Jason. But yeah, I think there's some of those little primnoid whips, and I think there are actually some bryozoans back there as well. And those look a little bit like corals sometimes. They have kind of that structure of a coral kind of branching.
How much time do we have left?
Sorry, Rian, there was, but in between
40,
that, there was something else. It was
5.5 minutes.
It's kind of transparent and delicate. And I couldn't tell if it was an octocoral or a hydrozoan. Just behind the sponge.
Yeah, interesting. Lots of these sponge skeletons here as well, kind of falling down from above. There are obviously some large sponge forms here at one point. And we've seen a few today. Another one of those parapeggers crabs. Seems like every ledge we look under, there's one of those hermit crabs today.
Oh, there's another one of these, I can't remember what they're called. You can snap it just for an ID. We've already seen this one. But
oh, one of these, yeah, benthic jellies. It's the second one of those that we've seen today. Beautiful.
Medico video. I'm flying by. I'm going to go for the larger subject right. Just have my curiosity. This one.
Benthicodon, like. Thank you, Scott. I was frantically looking through my notes. This looks like one of those large barnacles on the top of a, looks like an old coral stalk that's been crusted over by ferromanganese, so it's got that black-brown coating. I can't see if this barnacle is still alive or whether it's grown over by hydroids. It's kind of got some interesting projections out of it.
Video can come in partial.
Oh no, it had its feeding legs out. Now it's sensed our presence and has tucked them in.
That's a big barnacle.
This is a great shot too, because this really shows those plates that we might be finding that are ferromanganese covered. And there's hydroids all covered over the plates of this gooseneck barnacle, so it has a little neck there for the barnacle.
Could probably start stowing my arms.
Copy that.
Some forams. There's a little anemone in the top as well, and a crinoid. Lots of organisms. As Scott is pointing out, this is probably quite an old barnacle, just with the amount of encrusting that's gone on its outer shell there. What an amazing little community, all living together.
I'm going to zoom out and acquire lasers video. Let's get a scale for this guy. It's pretty big.
As we back out here, you'll be able to see two laser dots. These are harmless lasers that we use to scale things on the seafloor, and those lasers are 10 centimeters apart. So that barnacle itself is about 5 centimeters, which is really quite large for a barnacle.
Okay, you can come wide video, and I'll get our last views of bottom as we set up to take off. Video if you want to try to go for the fish. Not sure which way he's going, but
We have another fish up ahead. We've seen a few fish today, which has been great to see some more mobile fauna. It's quite far out. This looks another snappabranchid, I think.
Take more zoom if you got it.
And Ken Sulak was noting earlier that there's at least 2 species of snappabranchid that we've seen today. And both of them are relatively rare, and both of them, the observations that we've made today would be new depth records for the Atlantic, so that was good. You know, he was actually asking about this third one that's even more rare, but he would expect to hear that had this lateral line of very thin pores, and I wonder if that might be it. I don't know enough about fish ID to know if this is significantly different from the one we saw earlier, but this imagery is going to help.
Yeah, I was thinking the same, Scott. This white line, the lateral line going down its back with lots of these little white pores.
The floor and you can turn port. Yeah, port, and I'll go starboard a little bit.
Okay. Next video. Let him go.
Ken Sulak is a retired fish biologist from USGS who's done an awful lot of work with deep sea fish ecology, and he's providing us with just some outstanding information on the ecology, feeding ecology, and distribution of these
Watch
fish.
the pilot.
And so look forward to his report on that later.
That marks the end of our dive today. We're going to set up to come off. Thank you very much. All right.
And there we are. We ended on a potentially rare species of snappabranchid fish. This is the end of our dive today. I want to thank you for joining us on Dive 16 of the NOAA Ocean Exploration Expedition, North Atlantic Stepping Stones. We ended our dive today at just over 3,000, just under 3,200 meters. And certainly had a really exciting dive today, seeing some very unusual and very unexpected things, both to the geology and the biology. So on behalf of myself and everyone aboard the Okeanos Explorer, as well as all of our scientists on shore and our co-lead, Jason Chater, I want to say thank you for joining us. And also want to have a huge thank you to the pilots today and to the crew of the Okeanos Explorer. This has been a tough dive in terms of currents, and so they've done a great job keeping us on the seafloor today. So thank you. I hope you will join us again tomorrow when we hope to be exploring the Atlantis II Seamount.
Pushing out of depth.
Thank you, everybody.
Thank you, everybody.
Thank you for making that look so easy. We learned a lot today. A lot of surprises.
Bridge ROV nav.
Lots to chew over in the coming months.
The vehicles are off the bottom.
Full down and back. Good Delta Z.
Thank you.
Oh, if I mean, oh, you're already doing that. The old Titan.
Looking for you.
Pushing aft. Okay. You can take out your auto heading.
Copy, coming out.
I can see in my tail camera that I'm pulling the tether tight.
Sounds good. Yeah, sorry, I can't find you in the Titan at the moment.
It's alright. If you find your light pool, I'm probably in that.
Going to light pool.
That's forward.
So I'm going aft here. There we are. There you are.
Okay.
Yeah, looking good.
Killing my Z-bias. Taking out my heading. Your auto heading's already out. Dry locking a little forward way here.
I'm gonna bring down your aft bar and poles. Dial up your augs a little bit.
Okay. Alright, you can start hauling in on the winch.
Copy that. Easing up. Coming up.
Alright. Dry locking some forward way. Pushing up 60%. Delta's 3. I'm going to aim for a delta of indicated 20.
Copy that.
Looks like you're dialing in 28. Delta is 19. 819. I think I'm pretty close. 18, 19.
Copy that.
How you doing over there?
We're just about 30 right now.
Getting her dialed in, nice.
I think that should do it.